Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan selamat datang di acara Multiple Approach of Advanced Cancer Therapy and Handling Cytostatic Drugs Workshop. Senang sekali kami bisa berjumpa kembali dengan Bapak Ibu pada pagi hari ini dalam keadaan sehat untuk melanjutkan acara Wahid Hasyim International Conference on Advanced Pharmaceutical Sciences 2022 Multiple Approach of Advanced Cancer Therapy and Handling Cytostatic Drugs Workshop.
Good morning. May peace and blessing be upon you all. We are happy to see you all again this morning in good health to continue the event of the Wahid International Conference on Advanced Pharmaceutical Science 2022. Multiple approaches of advanced cancer therapy and handling therapy drug workshop.
Para peserta yang terhormat, agenda seminar hari ini adalah sebagai berikut. Sesi pembicara pertama akan dilaksanakan pada pukul 8:15 waktu Indonesia Barat. Agenda selanjutnya kami persilakan para peserta untuk dapat rehat kopi. Sesi pembicara berikutnya akan segera dimulai tepat setelah rehat kopi pada pukul 9:30 waktu Indonesia Barat. Sesi presentasi oral akan dilaksanakan setelah rehat kopi. Sekitar pukul 12 waktu Indonesia Barat kita akan istirahat makan siang. Pemaparan materi akan dimulai setelah istirahat makan siang sekitar pukul 13 waktu Indonesia Barat. Kemudian pemaparan materi terakhir direncanakan pada pukul 14 waktu Indonesia Barat dilanjutkan dengan penutupan.
To all session will be held after we will have a lunch break. The presentation for the lunch break will start after the lunch break around p.m. The third presentation session will be held after lunch break about 2 p.m. and the agenda we will gather paid in this room for closing ceremony at the end of the day.
Dimulai dengan sesi pertama adalah pemaparan materi dengan tema Good Manufacturing Practice for Clinical Trial of New Anti-Cancer Drugs oleh Asosiat Profesor Dr. Farahidah Muhammad. Sesi ini akan dipandu oleh moderator Ibu Apoteker Malinda Prihantini, Magister Sains. Kepada Ibu Apoteker Malinda Prihantini, Magister Sains, dipersilakan.
Let's begin to the first session with the presentation with the theme Good Manufacturing Practice for Clinical Trial of New Anti-Cancer from Associate Professor Dr. Farahidah Muhammad. This session will be moderated by Mrs. Apoteker Malinda Prihatini, Magister Sains. To Mrs. Apoteker Malinda Prihatini, Magister Sains, the time is yours.
Tes. Oke. Tes. Oke. Tes. Oke. Mrs. Farah, can you hear me? Can you hear me? Mrs. Farah? Okay. Okay, I will begin. Ladies and gentlemen, how are you today? Fine. Okay. Are you ready for the next incredible experience today? Yeah, I hope you so. Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Okay, I am very glad to see you again in this conference today. Yeah, let me introduce myself. My name is Malinda Prihantini. As your moderator today, I will guide you this session. We will listen to the presentation from our invited speaker, Associate Professor Dr. Farahidah Mohammed, with the title of the lecture today is Good Manufacturing Practice for Clinical Trial of New Anti-Cancer Drugs, the Conventional versus New Generation Dosage Form. Associate Professor Dr. Farahidah Mohammed currently is a Deputy Director Innovation and Commercialization, Unit Research Management Center in International Islamic University, Malaysia. She obtained her Bachelor of Science in Pharmacy from University of Strathclyde, Scotland in 1999, and then continued to work as a pharmacist in community pharmacies and government hospital in Malaysia for about 5 years before she took her PhD in Strathclyde Institute of Pharmacy and Biomedical Sciences in Scotland and graduated in 2008. She has been leading various research projects funded by various agencies and ministries in Malaysia. She had also received industrial grants to lead and coordinate GMP-associated training. She had participated in more than 30 international conferences. She has more than 30 publications and had supervised for more than 17 postgraduate master and also PhD students. She also received various awards and recognition, as in 2018, she got an award for 15 years of service from IIUM Malaysia. Her area of specialization is biopharmaceutical, biotechnology, engineering technology, applied science and technology, nanotechnology, medical and health sciences, drug discovery, drug delivery system, and also gene therapy. How incredible and inspiring, right? Yeah. So as I saw you very curious and maybe you have been looking forward to our invited speaker. Yeah, I will continue to the next session, but before we continue to listen the presentation from our invited speaker, may I tell you about how the presentation will be going on? This presentation will be split in two sessions. The main session is the presentation from our invited speaker, and then followed by the question and answer session. And then I will close the session with the conclusion. And so now I will allow to welcome our invited speaker, Associate Professor Dr. Farahidah Mohammed, to deliver the presentation. Dr. Farahidah, please enjoy your session. Thank you.
Bismillahirrahmanirrahim. Is my voice loud and clear? And this thing can be audible?
Yes, it's clear enough.
Bismillahirrahmanirrahim. Allahumma salli ala sayyidina Muhammad and اللهم صل على سيدنا محمد الفاتح لما أغلق والخاتم لما سبق والهادي إلى صراط المستقيم وعلى آله وصحبه وسلم. Alhamdulillah, thank you very much for inviting me to this platform to share with you some of my recent experience with good manufacturing practice. Okay? For today's topic, I have chosen good manufacturing practice for clinical trials of the extract. Basically, it's going to be introduced to you the concept of GMP and the drug development for conventional research new generation dosage form, which actually we call it as advanced therapeutic medicine products, ATMP. I'm sure all of you already heard about ATMP. Okay? Before, now the slide is not moving. Alright, so this is the outline that I will walk you through with my lecture today, with my speech today. I'm going to start with some just refresh with you on the conventional dosage forms and then next generation dosage form, which is actually they are now called as one-time medicine products. We are going to look at this particularly the group of what we call ATMP. Okay, and then I will share with you the draft and the timeline. Right. So after that, I'm going to share with you the introduction to good manufacturing practice. Okay, good manufacturing practice actually is a huge topic by itself. Everything in 1 hour time. So that's why I have to mention it is a very fundamental or introductory on GMP. Okay, for this session. Yeah, I'm going to walk you through with the conventional, ATMP, GMP. And then I will summarize my final slide. Alright. Now the conventional dosage form, I believe all of you are very detailed with what we call as conventional dosage form. These are the dosage forms from various classical dosage forms such as the ointment, such as the formulated tablet, the solid dosage form, the capsule, and the syrup, the liquid form, suspension, and the semi-solid, whereby we have the lotion and cream, okay, and ointment. Okay, the conventional dosage form, we formulate them using usually a single chemical-based compound. We call it pharmaceutical ingredient, right? It can be from small molecular drugs, which are acidic or base drugs. Okay, it can also be extracted from herbal or from the plants, where sometimes molecules and plants should be pharmaceutical grade herbs. We need to characterize the special species and then take out or identify the all is from the Whicaps species. There is one species that really can express the highest biomarker, which is 1,8-cineol. If it is a pharmaceutical grade, it expresses as more than 70% based on USPBP specification. Okay, so these are still under the conventional dosage form, where I formulated them very basic as an ointment. Okay. It can be manufactured under the non-sterile or sterile manufacturing condition for conventional dosage form, depending on the route of drug administration. So for the conventional dosage form, another criteria is that they have a common product spec according to the type of dosage forms, which mentioned in pharmacopoeia of USP or BP. Indonesia has their own Indonesian pharmacopoeia, so you are well advanced than us Malaysia. Anyway, so the next generation dosage form, as opposed to the conventional dosage form, are all these nanoparticles or microparticles, as illustrated here. Okay, we have mesoporous silica nanoparticles, or it depends on the size. And liposomes, we do have millimeter size, micron size, and nano size. And liposome evolution is also very fantastic. They have now transferosome, they have now ethosome, they have now neosomes, etc., etc. Okay, for the liposome. And then we have dendrimer, we have solid lipid carrier system, polymeric micelles, hydrogel, where we incorporated polymers in it. And then my cell itself, polymeric nanoparticles, gold nanoparticles, carbon nanotubes. We have used a viral carrier system, attenuated viral vector system, okay, and nanogel, nanocage, quantum dots, and amphiphilic cyclodextrins. Okay, these are all next generation dosage form. Okay, having said that, they look very simple like this, right? Very illustrative. But the final dosage form, visually, what we can see is only a powders. Okay, it can be a gel, it can be powders, or it can be even only a liquid. Okay, which is actually incorporated with other excipients to stabilize and to maintain the shelf life, required shelf life for all these next generation dosage form. So for today, we are going to share with you, we are to learn about the ATMP, okay, advanced therapy medicine products, whereby we have cell-based or genetic-based or gene-based advanced medicine products, whereby this kind of molecules it can be used or can be formulated as mesoporous or can be formulated as liposomes, as these are the carrier systems. Okay. Right. For example, here, these are the microspheres and nanospheres particles formulated by polylactic glycolic acid, a biodegradable polymers. Okay. These are my own PhD research last time in 2007, 2008, I graduated. Okay, these are just to compare and show you. These are the size of microsphere, and the small ones here are the were the nanospheres. So the size, as you can see here, is very huge difference between the micron and nanoparticles. So any advanced therapy medicine products, ATMP, like cell-based or the gene or DNA RNA based, they can be encapsulated inside the microspheres or nanoparticles. And it depending on the method of fabrication, we can choose, we need to do a formulation or check various parameters to ensure that we have encapsulate and achieve a good or sufficient encapsulation efficiency of these cell-based or the gene-based drugs. Okay, so these are just one of the examples. Now, moving on the advanced therapy medicine products, ATMPs, they are really innovative and complex process of manufacturing. Okay, they depending on type of ATMP, we can only source of the raw materials themselves very scarce, very rare, and very specific. So it depends on the manufacturing method and it depends on the type of ATMP. Every single of them will not have a strict or a fit one specification in USPBP. They are very different from existing conventional drugs, which is the small molecule drugs. If, for example, of ATMP stem cells for cancer that have been used to treat cancer or can also be used for regenerative medicines. They pose challenges to the design and conduct of clinical trials because it is very difficult to think about what is the placebo would like that should be in the clinical trials. Okay, even phase 1, phase 2, phase 3, and phase 4. And in terms of manufacturing difficulties, the type of manufacturing of these advanced therapy medicine products, such as stem cells, we actually need a not really huge facility of the manufacturing, but again, the required specification for the GMP setup or the premise itself should be sterile or aseptic preparation. It depends on the stability of the stem cells or the cells that we are manufactured. Okay, it still will cost us huge investment in terms of shelf life. They are very, very critical, and there will be a very tight control on logistic parts. Logistic, we are referring to the manufacturing, the way we store store, handle, and transport our products. Okay, I will show you towards the end example of an approved ATMP, approved by FDA for use. Okay, and this kind of ATMP, they also has a very different than the normal in normal conventional products, whereby during the writing of the method and protocol of the clinical trials, we need to have a long-term follow-up methods or protocol to be in place in the write-up of the clinical trials. And actually, usually it is very not always feasible to produce or provide preclinical data when we want to do a clinical trial. We need to apply ethical approval, right? So in one of the documents that we need to submit for ethical approval is the preclinical data, at least on the mode of action of the advanced therapy medicinal products that we are going to do the clinical trial on. Okay, so it is quite difficult, and even they are difficult, we still need to produce some proof and some risk assessment to allow us not to give a preclinical data, the animal study, I mean. But of course, for ATMP, there are a lot of various models to employ any in vitro cell lines or any in vitro environment to test the drugs. Okay, before we move on, just to ensure that we have a same understanding on the same page, there are 4 classes of ATMP, whereby we classify them as gene therapy medicine products, GTMPs, okay? Somatic cell therapy medicine products, SCTMP, tissue engineered products, TEP, and we can also have a combined ATMPs, combination of GTMP, CTMP for tissue engineered products. We can have a combination of ATMPs. So there are 4 classes of ATMP. Under the ATMP for cancer, usually the scope that people have done or research on is gene therapy medicinal products, GTMP. There are various approaches where we can do the gene silencing, we can do the antisense or create antisense therapy. We can also target and design the RNA interference and do gene and genome editing. And we can also have somatic gene therapy. Other people have also done somatic cell therapy medicinal products. If you key in or Google ATMP for cancer, for example, or ATMP as generals, there are a few other ATMP molecules that can be used for regenerative, for any injury, tissue injury, for liver disease, etc., etc. Okay. All right. So these are the normal timeline for R&D of drugs, from the drug discovery until it is being developed and approved for market use. Okay, I believe all the participants have came across this similar drug R&D timeline, whereby for the discovery phase it could take 2 to 5 years. Okay, and development phase can take another 5 to 10 years. Especially for the ATMP or the next generation dosage form, we can actually expedite the things like COVID-19 vaccines just now, because they have done various researchers have done the early discovery, have optimized their design of carrier system. So based on that, it's like a puzzle, we can match them, and finally we can fasten the drug development until it is being approved for human use. So from this discovery process, I would like to actually highlight this where the need of GMP is required. Okay. So before we GMP is required, of course, during the discovery or the screening process of target phyto compounds, for example, or target gene, etc., until they finally meet a lead compound to a candidate compound. Okay, they tested in animals, right? So these are the phase where we call as preclinical development or preclinical stage, where we tested in rats or in higher primates like monkeys, like dogs, etc. Depending on the type of diseases we want to treat. Okay, even for the preclinical study, we need to have a quality data, whereby the data or the instruments that we use should be based on DLP principles. Okay, what is DLP? DLP is good laboratory practice, right? This is to ensure that we obtain a reproducible data, and the data is traceable, and also the data is reliable. So all this concept of good manufacturing practice, good laboratory practice, and good clinical practice, they are all with same aim, which are to ensure we obtain reliable results and reproducible results and traceable results or data. Okay, for the GMP itself, regardless whether they are conventional drug formulations or they are advanced therapy medicine products like stem cells, okay, both of them will require preparation or manufacturing in a GMP environment. So before we start the phase 1 or the first time trial in humans, which is the phase 1 clinical trial, we need to find where actually a GMP facilities that can produce for us the stem cells, for example. Okay. And before we do that, everything here need to be specified in the document or meant for clinical trial, method or protocol of clinical trial itself. You need to ensure you know the GMP facilities, or else you need to apply for a grant to set up these GMP facilities. These are for new testing of a new stem cells, for example, stem cells or any other advanced therapy medicine products. Okay, you need to be a competent person in GMP as well as GCP, good clinical practice, in order for you to write a good protocol. Okay. This good clinical practice protocol is a blueprint of your studies from phase 1 and the next study, which is phase 2 and phase 3. Okay, it's a blueprint whereby all centers, if you're designing a multi-center clinical trial, all centers will use the same protocol. Okay, so for the GMP, it will require for phase 1 clinical trial, it will require the samples or the drugs that you want to test in human. It will require a preparation in GMP environment. So what is actually GMP? Okay, this is what I mentioned just now: good manufacturing practice, good clinical practice, and good laboratory practice. Okay, each of them has their own guidelines. And each of them, like good clinical practice, we have main authorities that actually develop, implement, and monitor the compliance on these guidelines. We have good manufacturing practice, good clinical practice, and good laboratory practice. Usually, the main authorities are from US, Europe, both of these countries. Okay. For the good manufacturing practice, of course, Japan have their own, and Russia have their own. However, most of the documents are actually harmonized documents. Okay? They share each other, and US FDA has the longest history on setting up or developing all these guidelines. Okay, for today, I'm going to walk you through with good manufacturing practice guidelines meant for conventionals and also for new advanced therapy medicine products such as stem cells. Okay. GMP is a concept that ensures products are consistently manufactured based on a controlled procedures. Consistently has to be proven based on a validation study. So this validation is another area of GMP, which is huge things that will spend a lot of times and a lot of funds as well for validations to prove that they are consistently produced or manufactured based on a controlled procedures. Okay, control procedures is how to control the procedures is actually based on an approved procedures. Not only GMP but also GCP and the GLP. They have a procedures that need to be approved first by superior. Okay. When talking about procedures, of course, there are initiation of the procedures or SOP, if you would like to use the word SOP. Okay, initiator is there, and then we send to a reviewer or two reviewers, and then if the reviewer asks to revise, go back to the initiator revise, and then go to review again, and finally approve. So these are the control procedures that will be used for routine manufacturing. Okay, under a control environment, how can we ensure that the environment is being controlled? Of course, there are strict specification of environment, and it has to be monitored every day. I will share with you who are responsible for the control environment. And finally, the results of this consistently product control procedures and control environment, we would like to see a consistent product quality being produced by a factory that have a GMP certified or a laboratory that has the GLP certified. Okay, based on appropriate quality standards. Okay, appropriate quality standards actually referring to quality standards set by the if there are drug products such as paracetamol, which is everyone I think know what is this API paracetamol. The quality standards of paracetamol need to be checked or cross-referenced in USP or BP. So these are what we mean by quality standards. It is not the quality standard we created ourselves. However, for new cancer drugs, for example, or new plant extract, for example, we need the researcher themselves, they need to develop their own monograph of quality, okay, based on the parameters set in USP BP. For example, identification of this new extract, plant extract, or the stem cell. What is the identification spec looks like? What is the protocol? Then what is the impurity level? And what is the, for example, the assay parts, the biomarker that should be there, and how much is the percentage of the biomarker? These are the quality standards that we need to develop along the research process. Okay, for GMP, there are source of references for these guidelines. Okay, the one that we usually based on is the PICS guideline, Pharmaceutical Invention Cooperation Scheme. Okay, PICS guideline, where many countries are the members of this PICS. And then PICS actually they also adopt some of the ICH guidelines. ICH guidelines is the one that having the long historic development process, and they have a very detailed guidelines even for the formulation parts, okay, and the analytical parts. Everything is in the detailed parts is in the ICH guidelines. And we also have ISPE, good engineering practice, meant for how we control the environment here. Yeah, and then we have USP, CGMP, EU, etc. Right. So this is example of PICS. Everyone who involved in GMP, they will know where to look at or where to find the guidelines. We can just quickly go and Google PICS, okay, and this thing will appear. And under PICS, there are several guidelines, including PICS GMP guide introduction, basic requirement part 1, part 2, etc. etc. Okay, if you can see here, there are dash 15, 15, 15, right? So the latest one is now after some revision, and they usually revise almost every year. And if there is a need for a new drugs like ATMP just now, they are very new. So they come out with a new guideline, and the revision will bear dash 60. Okay. All right. For GMP scopes, these are very, very fundamental GMP scopes. The one that I give you today is very fundamental. Okay. We need to have, these are very fundamental and critical one: quality control, production, engineering, and quality assurance. Okay, what if the GMP quality control do? They conduct testing on raw materials. Okay, meaning that when the raw materials that we buy arrive at the warehouse, we need to test raw materials. Okay. Similar to the ATMP, the raw materials need to be tested to know that they are correct raw materials, to know the impurity status, to know the the microbial limit of the raw materials, etc., etc. Where we can find the specification of this quality is from the USPBP or Indonesian Pharmacopoeia. Okay. And this quality control department, they also have to check on the quality of packaging materials and also finished product. And not only that products, they also responsible to check and monitor quality of the water used in the manufacturing site. Okay, water is very, very important in the manufacturing area. They involve in the product itself is the water is used in the manufacturing of the products. Or if not, even if not, they are used in the cleaning of the equipment equipment of the manufacturing in the plant. Okay, so water quality is very, very important. And not only that, because this is GMP, and they are when we talk about GMP, of course there is a clean room. Okay, the clean room, depending on the grades of the clean room, A, B, C, D, they have a different air quality. So that air quality is being produced using HEPA that can filter 99.99% of particles out. So everything is clear. And the airflow inside the room is also controlled. So quality control department will ensure some of the specification, especially on the microbial microbial content in the air quality itself. Okay, so these are the GMP or GMP for quality control. Next is production department. They are the one who knows or competent how to operate the machines, who knows and competent to maintain the machine and to clean. For the cleaning of the machine also has to be based on a control procedures. Okay, meaning that the protocol or the SOP already been initiated, reviewed, revised, and approved. So the production people, they manufacture products according to an approved process, and some of them will conduct the in-process quality testing. Okay, for the engineering department, they are also very important people. Okay, not many pharmacists graduate can involve in these GMP engineering departments. We usually recruited, or we usually recruit people from engineering background, because they would know how to maintain the engineering parts of the premise of the factory. Two main utilities involved in GMP is water purification system, like our purified water filter at home from Coco, from Amway, Kowe, etc., etc. There's so many brands. These are the one that we use in the production area is many times more higher volume and higher spec than what we have at home. Okay, the water purification system is very important to be maintained and ensure the quality of the water come out as a fulfill the specification in the USP and BP. Okay, another utilities important one is the HVAC system, ventilating and air conditioning, heating, ventilating and air conditioning system, HVAC system. These are also very important. And then the engineering people, they also need to monitor quality and status. So you can imagine that when we talk about GMP, these are all the basic GMP's premise or organization that should be there. Regardless whether they are used to manufacture conventional products and also the cell-based therapy or stem cells, etc., etc. So these are the basic ones. And finally, we have these quality assurance people, whereby they will monitor all the documents in and out, and they will also manage and manage the quality risk management of the whole GMP procedures, and they involve in regulatory requirement as well. So quality assurance is like an inspector where they inspect people whether production has done correct way, follow the protocol properly or not. Production people and engineering people has done it based on approved procedures or not, etc., etc. Okay, these are the quality assurance people. Okay, so I would like to share with you an example, a very quick example on iCool. iCool is an ointment whereby the iCool is the conventional products. So basically, we have these raw materials of iCool, we receive, and then we do the raw material testing. Okay, so the production process is based on approved PMR and BPR. PMR stands for batch manufacturing record, and BPR stands for batch packaging record. Okay. So production in GMP factory, we have a protocol or step that known as perform line clearance, and then dispense raw materials, and then we need to print and label properly, and then transfer to manufacturing room, and then start mixing process, and we need to do IPQC test. Okay. I hope my screen is okay and the diagram is large enough to be read. Okay, and then we need to discharge the next process, discharge the molten ointment into a tank and start to fill. Okay, collect and weigh all the waste and form IPQC again, and finally transfer of iCool ointment. Then after we have finished products, we have to do this kind of QC testing. These are all based on BP and USP for ointment. Okay. All right. That's it all for the GMP for the conventional. Now we move on for GMP for advanced therapy medicinal products, ATMP, which is the next generation medicinal products. Okay. Similar to the conventional, PICS now has revised their GMP guideline where they incorporated two new things, annexes. Okay, for the GMP guideline or other guidelines, especially for GMP, okay, and also in Malaysia, we have drug registration guidance document. These are another thick book where we need to follow all the registration requirement if you want to register new products, medicine category or cosmetic categories. Okay, we need to follow. I'm not sure, Indonesia, I'm sure you also have a drug registration guidance document. Okay, in that guidance document, the important parts, not only the first, the guidelines, but also all the annexes or appendices. Okay, there are very important parts where the regulators usually put all the formatting, what kind of things in details form that need to be submitted. So in this case, for PICS GMP guide, they have an Annex 2A. Okay. To fulfill or to provide a GMP guideline for advanced therapy medicine products for human use. Okay, these are very, very relevant to what we are talking here about the stem cells, about the new therapy for cancers. Okay, so we need to be based on the manufacturer, the Annex 2A of PICS. Okay, here is the revision they have made. If we go to the PICS guideline, you will see this -16 here. And we need to check the annexes. The annex to A is in this GMP guide related annexes. Okay. So these are the excerpt from the annex to A here. All right. So the GMP requirement is based on manufacturing method of ATMP manufacturer. For example, if we are now working on gene therapy ATMPs, there are genetic modifications that can be obtained through a variety of methods, whereby we have viral and non-viral vectors, we have mRNA, whether it is ex vivo, in vivo gene editing tools, etc., etc. These are very, very specific process and a longer process, much longer processing, manufacturing as compared to the conventional. Okay. The genetically modified cells can also be from human origin. So these are the raw materials involved in the ATMPs. As compared to when we make an ointment just now, cool ointment, we have the eucalyptus oil, we have the ointment bases, okay, we have other oils such as menthol or peppermint oil. So these are the conventional dosage form raw materials. But for ATMP, the raw materials start from these modified cells or the gene sequence that we are working on, that we want to amplify in the manufacturing environment or scale up. Okay, and it can be it depends on the intended use of the ATMP, whether it is for autologous use or allogenic use. Autologous means we take out a cell from a body of human A. We do something with the cell to, for example, to remove all the bad things, and then we incorporate or inject back the good one into the same body of the people. That is autologous. Okay. For allogenic, there is a donor of the cells, and the recipients of the cell is a different people. That is allogenic. Okay, in a medicine product, the genetically modified cells or gene therapy products can be presented alone or combined with medical devices. So the manufacturing of ATMP will be very, very more complicated than the conventional products. We need to incorporate whether the finished product that we want to present is the gene in simply a polypropylene bag, or it combines with a special medical device, okay, for implant, for example. Okay, in the Annex 2A, they mention this. They give an example. As you can see here, there are various types of ATMP. Okay, it can be from the gene therapy base, or it can also be from the cell therapy base, and also tissue engineered products. There are two main categories: gene therapy, the somatic cell therapy, and tissue engineered products. Okay, so these are all under category of ATMP. Okay, if you can see here, the one in green, all right, the darker green here, is where the GMP is applied. It requires a GMP protocol and GMP environment. Start from the raw material testing, start from the environment that is controlled, the water that is controlled, water quality that is controlled, etc., etc. Okay. Right. For example, here, I'm sharing with you an approved somatic cell therapy medicine products for cancer. This is an example for approved by FDA for human use. Okay. The brand name is known as Provenge. It has this Sibuleucel T as the cells that they cells, and also the excipients that they use for the autologous cells. As you can see here, it is only meant for autologous cellular immunotherapy, whereby we have to take one cell from single human, do something with that cells, and then incorporate in their formulation, and then re-injected into the same human. Okay. So these are the immunotherapy indicated for the treatment of asymptomatic or minimally symptomatic metastatic castrate-resistant prostate cancer. Okay. So each dose of Provenge contains a minimum of 50 million autologous CD54 54 plus cells. All right. And it is being activated with this PAM, GM, CSF. All these are the fluids that maintain the stability of these CD54 plus cells. Okay, as mentioned again, these are autologous, meaning that they take the CD54 cells from the same human and incorporate back after they do something with this, and they amplify these good cells. They re-inject a huge amount of the cells. Okay, 50 minutes. Okay, the recommended course of therapy for Provenge is 3 completed doses, approximately 2-week intervals. These are what they have studied during their clinical trials. Other than that, they did not study. So what they studied during the clinical trial is the things that they need to write down in their package insert. Okay, for registration, we need to come up with our package insert where we have titles like the recommended dose, the indication, and the adverse effects, etc., etc. Okay. And this Provenge is designed to stimulate the immune system. So with the higher immune system, the body can fight the recurrent prostate cancer that this body have. Okay. For this, the somatic cell therapy medicine products, they have in their package insert, as I mentioned, it is for autologous use, so they need to confirm the patient identity. Okay, the Provenge is intended solely for autologous use, so the patient identity need to be confirmed before they can administer to the patient. The patient must bear the same name as the Provenge that we receive from the manufacturer. Okay. Okay. Right. And not only that, for all the ATMPs and also the biopharmaceutical products like vaccines, we need to have a product release confirmation. For this Provenge, the manufacturer, when they take out the cells from the body of say myself, okay, I have prostate cancer, and they take out my cell, and this cell, my own cell, will be propagated in their manufacturing factory, and when during the course of manufacturing, they will release once the product or the cells is ready, they will transfer to a medical or clinical facility for me to receive the cells. Okay, hospital, basically it's a hospital, or any hospital who actually has this kind of facility to deliver a cell therapy based medicines. Right. So when we receive this, we need to wait for the approval or confirmed product release from the manufacturer. The manufacturer here of the Provenge is Dendron. Okay, they will send us a cell product disposition form containing the patient identifiers, expiration date and time, etc., etc., and status whether this can be approved for injection or not. Okay. Why this happen? Because of the testing takes sometimes. The product already arrived at the facility to be administered to my body, okay, but the testing yet to be completed. So we just need to wait from the Dendron after they have finished their quality control testing. Okay. All right, this slide is showing you simplified manufacturing process flow for ATMP. Say, for example, we have a gene, we have a barren gene taken from a patient. Okay, and then we do something with the gene. For example, we cut the gene. After we cut and make a good gene, we have to encapsulate or transfer into a vector. Yeah. Okay, we can use viral vector or non-viral vector, which is normal plasmid. Okay, why we use a vector, because we need to protect the gene, and the way the gene give their manifestation on the disease itself is by, for viral is easy because viral can penetrate into a nucleus and do all the transformation, do all the messenger RNA and all the process until they have the protein that they produce expressed in the body. The protein itself that will be the therapy for the patient. Okay, so basically along this process, you can imagine that there are strict control. The gene transfection into the viral, the gene transformation into the cells, and then cell need to be amplified in a cell culture. Okay, these are normal laboratory use, but in the manufacturing process, we need a biofermenter. Okay, there are huge 100 liters of all liquids, all the nutrients meant for the cells to grow. And cells here, normally we use E. coli, the bacteria itself. Because E. coli or the bacteria, they can really amplify, within 24 hours, millions of them can multiply, okay? So these are the way we amplify the genes that we want, okay? Then we do the process, whether they will be formulated into nanocapsules or nanospheres or microspheres and any other carrier system. Okay, so this you can imagine that they will require all the MP. Okay, for the gene therapy, for example, here the plasmid manufacturing will not require the GMP, but the establishment of a master cells bank and the working cells bank, these are the cells bank. Some of them will be frozen and become the master cells bank, and some of them will be used for working cells bank. Okay, we call it cells bank, and then start to manufacture amplify the E. coli just now, and then purify and then extract and purify the gene, and then the gene will go for formulation and filling into. Usually filling into some medical devices or special medical devices or only vials. Okay, but these are the formulation and filling will follow the conventional way of GMP, such as filling process and such as start with formulation process, where we have these raw materials, the genes itself, we mix with the excipients, the buffer, liquid form, and then we sterilize if they can be sterilized. Or if they cannot be sterilized, we need to ensure all the preparation follow the aseptic condition. Okay. Right. Just to share with you some basic quality control for ATMPs. ATMPs or any cells or stem cells manufactured for exploratory or early phase clinical trials, phase 1 and phase 2, they are expected to be validated. Okay, validated here is a huge word for GMP. Okay, under GMP, we have a validation plan that we need to plan properly. And usually they need 3 consecutive batches from the same cells, from the same working cells bank. We need to do the manufacturing 3 times consecutively. Within these batches, we need to also produce a specific cleaning process of the equipment that we use. Okay, so all these 3 consecutive batches, they will produce at least sample number of 3, right? And these are sufficient for the under GMP scope. This is sufficient to ensure or to prove that the batch or the quality of the products is consistently produced. Okay. All right. So within this quality control, they also, as I mentioned, if it is need an aseptic condition or sterilization process, And usually we have this viral inactivation or removal. These are the things that need to be incorporated during the validation studies. Not only that, the effectiveness of disinfection methods should also be proven, okay? Referring to the cleaning of the equipment themselves. All right, I come to my final slides. For summary, GMP for conventional medicines versus ATMPs. Right. Along my talk today, I'm talking about all these departments, all the quality control, the production, the engineering departments, and the quality assurance, right? So these are all similar, common, critical departments for GMP in manufacturing the conventional as well as the themselves. Okay, so they are actually can be summarized into these 5 P's. Okay, which are the first P is people, where we need to have a competent personnel. Personnel is there to run the things, right? In GMP, we have the personnel. There are very important things in the GMP, yeah, or in the GMP organization. We need them to be trained well, to be assessed, to be competent, and they has to comprehend their roles and responsibility. And then another P is the process, where we need to, first of all, if we do not know the process, we of course need to study and research, optimize the process. After the optimization part, then we write down the approved process protocols. Okay, and the premise, it will be the responsibility of all, and especially the engineering parts. They need to ensure the cleanliness and the equipment is well maintained. Basically, all the calibration is there, calibration of the HEPA system, calibration of the water purification system, the devices that need to be used to monitor or to analyze things. These are all the things that need to be maintained properly. Okay, and then the procedures. Another P is on the step-by-step or routine procedures. The routine one is called SOPs. Okay, there are various types of SOP, hundreds of them in a GMP setup. And final P is the products, whether it is a stem cells products or it's a very basic ointment products. All of them need to have a clear specification. And if you are all the R&D, I believe majority of the participants are research people. Okay, if you want to develop a products that and you have a direction to market your products, approved for market use, you need to start from now checking all the regulatory requirements for your products, whether they are stem cells, whether they are plant extract or the medicine single compound. Okay, you need to develop your own specification of your raw materials, start from the extraction, and of your final dosage form. Okay, I think with that, I would like to pass the session to the moderator or maybe Q&A.
Is there any question? Yes, please. Okay.
My name is Sari from Stipat Semarang, a college pharmacy in Semarang. Morning. My question is, how do we determine the stability of ATMP product? Are there the differences, the stability study of ATM with the other biology product? That's it. Thank you.
Okay, Dr. Farhida, can you get the questions?
Yeah, the basic study.
Sorry, you are still in mute.
No. Can you hear me now?
Yes, we can
Alright.
hear you.
Thank you for
Can
the
you hear
question.
the questions?
Yes, madam.
Okay, okay, please.
How to know the stability of the ATMP products, right? It depends on the type of ATMP products. Okay, if let's say they are gene therapy, the stability data that we need to provide before we can apply for ethical approval for clinical trials, it has two specification. One is meant for clinical trial stability. The other one is meant for manufacturing or the routine market authorization stability. Okay. For the first trial in human, there is a little less stricter than the market authorized stability data. Okay. The first of all, we need to check the stability study guidelines, Asian stability study guidelines for other products. We can follow their basic requirement first. That is first. You need to ensure you have appropriate container closure system for your API for your ATMPs. Okay, this need to be defined first. Which container is compatible with your ATMP products? Okay, and you need to ensure you already formulated them in such a way that they are stable. So how to study this stability? You need to study at least 3 different temperature range. One is fridge temperature, one is normal 25-degree temperature, and maybe at accelerated condition, which is 40 degrees Celsius. But normally, of course, we need a lower temperature, fridge temperature for your ATMP. Okay, if you have followed that, the specification that you need to set or identify, which should include the impurity degradation products, okay, in your final product stability. You need to include the expression data, whether after you store in appropriate condition, in that temperature condition range, 25 degree or below than that, the cold temperature 2 to 5 degrees, okay, whether it is still stable, it can still be expressed with what you intend it to be. If gene therapy, you need to check whether the expression is still there. So these are basic things. And if they are formulated that have a specific particle size, you need to check the particle size, whether it keep growing or the particle size is stable. The pH, these are very basic things that you need to identify as well. So I hope that answered the questions.
Okay. Previous question. Is there in Malaysian pharmacopoeia provide about the monograph of the ATMPS? Because in Indonesian pharmacopoeia we do not have the monograph about the ATMPS yet. Yeah, it's not covered yet about the ATMPS. And how about in the Malaysian pharmacopoeia?
Right. Thank you, moderator, for the questions. First of all, Malaysia do not have our own pharmacopoeia. Unfortunately, we do not have. It's maybe fortunately as well, because I think the main reason is that our population is very small. We don't really have a group or the human resource that really that enough for us to convene and do and focus doing all these monograph things and come up with our own pharmacopoeia. Okay, So we just simply based on other authority like USP and BP. These are the major two pharmacopoeia that we use. And the regulators for our regulatory for pharmaceutical in Malaysia, they also use these two, BP and USP. The Indonesia has your own, of course. You have big populations, and Alhamdulillah, you're also very competent in this area. So, for me, it is okay for us to just adopt and base on other people's pharmacopoeia. And even the US, they also do not have. They still yet to they still yet to incorporate this ATMP specification pharmacopoeia. The one that have done this earlier is European Commission. They have formulated their own, even the good clinical practice for ATMP guideline. You can check on the internet. It is freely available. And they also have come up with the ATMP specification pharmacopoeia. Not really pharmacopoeia, but the specification related to it. Okay. Bro. And we can also check the USP and BP. They usually every year they have the revision made to their MVP, and we can check. We just open up the USB BP and check whether they already have or not. Okay. Mm-mm. Yes, true. If we, as you mentioned, there are various set of cells, and somehow very few just approved. So that's mean there are a lot of information is not there. Okay, but do not be afraid. You can actually develop yourself your own specification or your own monograph. You can follow what literatures have done. Literatures say, for example, they have studied gene editing. They study the characterization of the genes, right? Expression is there. The transcription efficiency is there. So these are the things that we need to incorporate in designing our own monograph for our own ATMPs.
Monograph, and then we can maybe we can discuss with the government, with the regulator for
True.
the specification. Yeah, maybe for the next time we can conclude it in our monograph, in our pharmacopoeia. Yeah.
True,
Maybe.
true. You can
Okay.
justify. You can justify based on the previous studies. Usually,
Okay.
regulators very sometimes not all of them already competent with ATMPs, right? You are the one
That's
that
right.
study your ATMP, so you are the one who knows better actually than them.
Okay, that's right. We get
They like the sharing of the experience and the sharing of knowledge with the regulators. Especially
that's
these are
right.
very new ATMP, very new.
Yes, we get the point because the researcher is the person and is the responsible person which is more understand about the ATMPS maybe
Yes.
yeah.
You are the one who multiply, you are the one who amplify, you are the one who use the real equipment to do the scale up, for example, right? So, and even the characterization, everything, you are the one who are the most competent and know about your ATMPs.
Yes. Okay, thank you, Dr. Farahida. Actually, in Indonesian, we have Annex 12 in Indonesian GMP. We call it CPOB, Cara Pembuatan Obat Yang Baik. In Annex 12, it's provide about how the biological production should be manufactured, but it's not clear describe about how the monograph. It just describe how the manufacturing should be provide. Yeah, something like that.
Yes, true. The
Okay.
guideline will stay as very simple and guideline, but we need to cross-check with the USPBP to check for the specification.
Okay.
The best thing is to familiarize yourself with single compound specification, like paracetamol just now. So paracetamol in USPBP, they have first is the identification test. So you need to use the same terms, identification test for your ATMP. How to do the identification?
Okay.
So these are one of the criteria, specification. Another thing is impurities. What kind of impurities your ATMP has? So that's why you need to familiarize first with the USPBP, the quality spec of a single compound. That will help you a lot to understand how to develop your ATMP specification.
Okay. Okay. We get the point. Thank you, Dr. Farhidah Mohammed. Is there any question from the audience? Okay, please.
Okay. Thanks for the amazing presentation for Miss Farah Ida. Thanks for the opportunity. My name Anggria Syafirga Masikinuladeni from Bancel Program of Pharmacy, Wahid Hasyim, Semarang. I have any question? How is the development of nanoparticle product in Malaysia? And how did the Malaysian government support the development of nanoparticle for research? And what trends of nanoparticle in Malaysia? Thank you.
Okay, please, Dr. Farhidah to answer the question.
All right. Thank you for your questions, brother. For the nanotechnology in Malaysia, our government, yes, they for the past years they have supported researchers on the nanotechnology progress or development. Okay, it can be from engineering side, biocomposites on the nanotechnology. It can also be for the drug delivery groups for the nanoparticles or nanotechnology. So far, what I can say, the nanotechnology is being the research still at the fundamental level rather than scale up levels. Okay, but for the cosmetic sides, the nanoparticles or nanotechnologies, the general terms would be nanotechnology. For cosmetic development, they are actually better than the pharmaceutical development on using this nanotechnology because of the strict regulatory control on the specification. So these are the major drawback for nanotechnology development for the pharmaceutical area. For cosmetics, they have done a lot more than us. For the biocomposite or the engineering parts, nanotechnology, they have also done a good job. But I still think that they are still under fundamental stage. Okay, the prototype stage and fundamental stage rather than approved products for market use. For pharmaceutical, of course, there's none that has entered approval stage for market use. Even for clinical trial, they are very rare that people have done until clinical trial stage for nanoparticles in drug delivery. Okay, but for engineering side, for other ICT side, I think that one is and the cosmetic side, they are more advanced than us in Malaysia.
Okay,
Thank you.
thank you, Dr. Farhidah Mohammed. Is it clear enough? Okay, thank you. Is there any question? Is there any question? No? Okay. Ladies and gentlemen, we finally come to the end of the session. Before the closing up, I will take the conclusion from what the speaker had been presented. Today's era of the drug development is switching from the NCI, new chemical identity invention, to the new generation of drug delivery form, which is the product we call it ATMP. And the ATMP manufacturing need the GMP, SMS, the conventional drug. The GMP, which is regarding 5 matter, which is people, process, premise, procedures, and also the product. Okay, I would like to thank to the speaker for the interesting presentation and to the participant, which is very active today. Finally, give the applause for the speaker today, Associate Professor Dr. Farahidah Muhammad. Thank you for your presentation.
Thank you, Madam.
Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Disampaikan terima kasih.
Deliver, thank you.
Berikutnya adalah rehat kopi. Bapak dan Ibu dipersilakan untuk dapat menikmati hidangan yang tersedia hingga pukul 9:45 waktu Indonesia Barat. Setelah rehat kopi, agenda selanjutnya adalah presentasi oral, di mana sesi presentasi oral ini akan dipandu oleh moderator Ibu Apoteker Ririn Lispita Wulandari, Magister Ilmu Biomedik, Ibu Apoteker Siti Setianingsih, Magister Farmasi. Kepada peserta presentasi oral dapat melihat ruang presentasi pada program book. Ruang 1 adalah ruang sebelah dan ruang 2 adalah ruangan ini. Setelah rehat kopi, peserta presentasi oral dipersilahkan untuk segera dapat menempatkan diri. Kepada presentasi oral secara online, dipersilahkan untuk masuk ke breakout room yang telah disediakan. Presentasi oral secara online akan dipandu oleh Ibu Apoteker Dewi Andini Kunti Mulangsri, Magister Farmasi.
For the next session, the attendees are welcome to coffee break. The next invited speaker session will start promptly after the coffee break at 9:45 AM. After the coffee breaks, the next agenda is an oral presentation. To participants of the online presentation, you are welcome to enter the breakout room that has been provided. That presentation will be guided by Mrs. Apoteker Dewi Andini Kunti Mulangsri, Magister Farmasi.
di TikTok ini
Tes, tes.
Will guide the course of oral presentation today. And before I will confirm or make sure you're present today, I will call your name one by one, okay, according to serial number. Number 1, Nika Natania. Number 2, Sobari. Number 3, Galih Hidayanto. Number 4, Ihsan Nurlaili Safara. Have you been in this room? Okay, thank you. Number 5, Eva Monica. Okay. Number 6, Farah Fadiah Anwar. Okay. Number 7, Tiaratun Nikmah. Number 8, Sifa Maulida. And next, Farida Maira Lokana. And then Yanca Anas. Mr. Yanca Anas, oh, okay, thank you. Okay, because all presenters are already in the room, next I will tell you the rules for the oral presentation. Namely, the oral presentation will be conducted in English, and each presenter will be called to the front to make a presentation, and based on presenter serial number. Presenters can presentation using a laptop and pointer that has been provided by committee and will be assisted by the committee to operate the PowerPoint. Each presenter will be given a maximum time of 10 minutes for presentation and 5 minutes for discussion. And if the presenter has finished the presentation, I will invite the audience to ask the question. You can raise hand and mention your name and institution. And when the time is up, I will stop the discussion and presenters can get back to your seat. Then I will invite the next presenter. And this event, the best presenter will be selected by the jury and will be announced by the committee at the closing ceremony. And the presenter will be awarded as the best oral presenter. Okay, all right. For all presenters, can you understand what I mean? Okay, thank you. Okay, then I will start the oral presentation. All right, for the first one, please, Mrs. Nikan Natania. Okay, you can come forward to make a presentation. The floor is yours.
Di sini apa di? Cek. Assalamualaikum warahmatullahi wabarakatuh. Hamdan wa syukranillah amma ba'du. First of all, let's thank to our God Allah Subhanahu wa Ta'ala who has given us mercy and blessing so we can gather in this blessing, please. Secondly, salawat and salam always be upon to our Prophet Muhammad Sallallahu Alaihi Wasallam who has guided us from the darkness to the lightness. And thirdly, I would like to say thank you to the moderator who has given me an opportunity to deliver my research with the title Anti-Hyperpigmentation Activity of Serum Combination of Apple Stem Cell and Niacinamide on the Skin of Male Guinea Pig Cavia porcellus. Operator, please. Introduction. Hyperpigmentation is a condition where there is an expansion in the synthesis of melanin on the skin so that skin become darker or the appearance of dark spot caused by continuous exposure to UVB rays. Hyperpigmentation can be overcome by inhibiting the enzyme tyrosinase. Hydroquinone can reduce the amount of melanin up to 90%. However, the persistence of used hydroquinone can cause harmful effects such as the appearance of erythema, skin irritation, exogenous ochronosis, and cataracts. Niacinamide can overcome hyperpigmentation by inhibiting melanin transfer mechanism. Swiss apple or Malus domestica bulb contain high phenolic and flavonoid compounds, resulting in antioxidant activity that plays a role in overcoming hyperpigmentation on skin. Swiss apple were selected to be developed using the plant stem cell method, which can be stored for a long time. Based on research by Mahmoud et al., the emulsion that was made from extract of apple stem cell, which applied to hyperpigmentation sufferers, can play a role in reducing melanin. Stem cells are able to improve the quality of cosmetic because apple stem cell or stem cell can produce to be safer because they are natural ingredients which have good effectiveness. Research question. First, does the serum combination of apple stem cells and niacinamide have anti-hyperpigmentation activity in vivo on skin of male guinea pigs cavia porcellus exposed to UVB light? Second, does the combination serum of apple stem cell and niacinamide have more effective anti-hyperpigmentation activity than single serum niacinamide? This study aims to prove the anti-hyperpigmentation activity of serum combination of apple stem cells and niacinamide on the skin of male guinea pigs. This is my method. First, hyperpigmentation on the skin of guinea pigs was induced by exposure to UVB for 5 minutes per day for 2 weeks. 16 guinea pigs with hyperpigmentation were divided into 5 groups. These groups include group 1 is a positive control of melanocream, group 2 is a negative control serum without active substance. Meanwhile, group 3, 4, and 5 respectively are serum niacinamide group, serum with apple stem cells and niacinamide, and the group that was not given any treatment, and group 6 is a normal group. This is my second method. Each preparation was applied thinly and evenly on the skin of the guinea pig. All treatments were given topically and carried out at night for 2 weeks. On day 29, a tissue biopsy was performed with Mason-Fontana staining. The percentage of area was calculated by image digital analysis method. And finally, it was analyzed statistically using the Kruskal-Wallis test and continued with the man-Whitney test with a 95% confidence level. This is my result and discussion. Anti-hyperpigmentation activity test on the skin of male guinea pigs cavia porcellus was carried out by giving UVB light induction for 2 weeks with an exposure duration of 5 minutes per day. After being exposed to UVB rays, reddish spots appear on the skin of the guinea pig's back. Based on the observation occurred before the study, the exposure the guinea pig's back skin 5 minutes per day to
for 2 weeks has been able to form melanin pigment in the stratum basal layer. The black color in the image shows the melanin granules in the epidermis. The results of Man Whitney show that there was a difference between the negative control and the treatment group that was given combination of apple stem cell and niacinamide. This indicates that the serum combination of apple stem cell and niacinamide has anti-hyperpigmentation activity. Meanwhile, the treatment group that was given the niacinamide serum was compared to a serum group with the combination of apple stem cells and niacinamide, which showed that there was no significant difference. The result shows that the serum combination of apple stem cell and niacinamide of apple stem cell had anti-hyperpigmentation activity that was indicated by the percentage of melanin granules which appeared to be less in number than the negative control group. Serum combination of apple stem cell and niacinamide has anti-hyperpigmentation activity because Swiss apple contain antioxidant compounds that can act photoprotective. The cell damage caused by the exposure to UVB rays. Apple stem cells are rich in phytonutrients such as carotenoid and flavonoid.
I'm so sorry for Mrs. Nikan. Time will run out for presentation.
Yes, please.
3 minutes again.
Yes, thank you. The Swiss apple also contains phenolic compounds that can regulate several signaling pathways, which can inhibit xanthine oxidase, which is known as a source of frost. They are free radicals and contribute to oxidative stress. And summary, anti-hyperpigmentation activity test on the skin of male guinea pigs cavia porcellus was carried by giving UVB light induction for 2 weeks with an exposure duration of 5 minutes per day to prove whether the serum combination of apple stem cell and niacinamide had anti-hyperpigmentation activity. The apple stem cell are useful in overcoming premature aging and hyperpigmentation on the skin. Meanwhile, niacinamide has been shown to inhibit the increase of melanin. The result shows that the serum combination of apple stem cell and niacinamide has no significant difference from serum niacinamide. Acknowledgement. Thank you for Ms. Risa Filafitria and Ms. Junvidya Herawati, and thanks to Pharmacy Fakulty of Wahid Hasyim University. Thank you very much for your nice attention, and I'm sorry if I have a mistakes.
Okay, thank you for your presentation, Mrs. Nikan. Next, ladies and gentlemen, are there any question? Any question? Okay. Yes, please.
Can you repeat it, Ms. Farah? Okay, thank you, Miss. Yes. The treatment given at night because to avoid sun exposure, to keep my research going on will be okay. Something like that, Miss.
Okay. Any question? The others? Audience? Okay, please.
Can you repeat that, Mr. Gali?
The committee,
We did.
please provided a microphone for. What's your name? Please mention your name.
Cek, cek, cek.
Okay. My name is Kelly Dayanto. We did it use Apple Swiss, Apple system cell from Apple Swiss.
Okay, why I use Apple Swiss? Because Apple Swiss have many advantage. Which one is Apple Swiss contain high phenolic compounds and carotenoid, and that can be reduce melanin of hyperpigmentation on the skin. I think enough, Miss.
Okay, enough?
Yes.
Okay, thank you very much. Please give applause for Mrs. Nikan Natania. Thanks for your presentation. Okay, I will be called the next presenter, Mr. Sobari. Have you been in this room? Okay, please, you can forward to make presentation. The floor is yours.
Ya, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah wa syukrulillah wa sallallahu ala Muhammad ibn Abdillah amma ba'd. The first I would like thanks to Pharmacist Rini Sifta as a moderator. Thanks, Miss Ririn. Dear Miss Malinda Pihandrini as our supervisor, and honorable our audience of this seminar. Okay, good afternoon, lecture and audience. First, let me introduce myself. My name is Sobari, and I from Fakulty Pharmacy of Hasyim University. Okay, I saw on the screen, there is a very large picture of red dragon fruit peel. And the title of my thesis is Application of Flex System in Oil in Water High Day Cream, Red Dragon Fruit Peel Juice, Hyloceres pylori juice with twin span and glycerin combination. From the picture and my title, we already know I have 3 points important of my research. The first is red dragon fruit peel. The second is natural dye, and the third is variation of emulsifier. Okay. The point 1 is a red dragon fruit peel. We have been consuming red dragon fruit as a fresh drink, like a juice or eaten directly. Of course, we leave the peel, right? The red color on the fruit and the screen and the peel is attract to me to use it. Why peel? Why we not use the fruit? We are know the fruit is better to eaten directly, so we use the peel. And Ramawati and Handayani and it 20 and 22 state in the peel of red dragon fruit is contain high of beta-cyanin and anthocyanin. Okay, the 2 is natural dye. What is anthocyanin and what is beta-cyanin? Beta-cyanin is one of color pigment we found in natural ingredient, and this can use to natural dye. Beta cyanin generally have bright color. Thus found in red dragon fruit peel. The tree is variation of emulgator. The preparation we choose of this application is a cream. In manufacture a cream, we have two emulsifier. We choose twin, span, and glycerin to handle it. The combination of three emulsifier is expected to increase the stability of emulsion. Okay, now what is the problem we are questioning? The first is, what are the physical characteristics? Oil in water preparation, hair dye cream, red dragon fruit peel juice, Hyloceres polyrhizos, with span, twin, and glycerin combination. The second is, how is the coloring ability of the cream red dragon fruit peel, Hyloceres polyrhizos, with span, twin, and glycerin combination? After we knowing the problem, what are the goals we want to achieve? The first is to know physical characteristic. The second is to know coloring ability. Okay, next slide is method. The first, the collection material. We choose material is red dragon fruit with family Hyloceres polyrhizos. The second is plant determination. This is to determine the variety use. Then is the processing, where the peel red dragon fruit is used to extract the juice. Dry to freeze dried method. We choose freeze drying method because beta cyanin is very heat resistant. Next is making the leg system is by mixing alucor, red dragon fruit peel, with aerosol in ratio 10, 9, and 1. Followed by making cream preparation and slowly added to be cream with leg system. The last process is evaluation. Evaluation is rate power test, adhesion, viscosity, pH, and calorie. Okay, we present an overview. There is a leg system. This is a preparation, and this is higher before coloring, and this is higher before coloring after 2 hours. From the picture we have now, the coloring is fed more from the preparation. Okay, this is a sample. I use preparation. Okay, this is table. From the table, we can conclude value is more from 0.05. They indicate that there is no effect of concentration variation. Okay, this is table of multivariate. Kemudian, then is summary.
I'm so sorry, Mr. Sobari. the time 2 minutes again. Okay.
Okay. Summary. Hair dye cream oil in water, the dragon fruit peel, has a good characteristic at each concentration and has no significant effect on the physical characteristic. Cream can stick well anyway, but tends to fade more from cream color. Okay, acknowledge. The author gives best gratitude to the research funding assessment from the Directorate of Research and Community Service, Directorate General of Research and Development of Ministry Research, Technology and Higher Education, through LLDT 6, Central Java, in the Beginner Student Research Scheme. This is a reference. Okay, thank you. That's all we can say. If there any mistake, I beg your pardon. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Okay. Are there any question for Mr. Sobari? Oh, okay. Any? Please, Mrs. Mention your name, okay?
Okay, my name is Sari from Semarang. My question is, in your presentation, you said that beta-cyanine is soluble in water, right? Yes, beta-cyanine is soluble in water. In your presentation, you said. So why you design the dosage form in the cream type oil in water? Is the beta-cyanine can include in oil in the design of cream type oil in water? And the second question is, you said in the last summary that the cream of beta-cyanine is tend to clearly in washing after washing the water, right? Why the beta-cyanine can stick well in the hair? Is the because the beta-cyanine is soluble in water, so it didn't stick well in the hair? Maybe you can explain the hair structure, maybe. That's all my question. Thank you, Mr.
Do you understand what Mrs. Ungsari? Yes. Question? Understand? Please, can you explain? Okay, not understand. Oh, please, Mrs. Unsari can reply your question.
Maybe I speak in Indonesian. Ya, Pak Burhan ya tadi. Sobari, mohon maaf Bapak, Pak Sobari. Yang pertama, pertanyaan yang pertama, di presentasi Anda mengatakan bahwa beta-cyanin itu larut air begitu ya. Salah satu pigmen warna yang memang menarik begitu, tapi dia memang sangat larut dalam air begitu. Nah, mengapa Anda mendesain beta-cyanin itu ke dalam krim tetapi pada tipe minyak dalam air begitu, tidak air dalam minyak? Apakah beta-cyanin bisa masuk ke dalam globul minyak dalam air begitu, dan apakah itu juga alasannya di akhir Anda menjelaskan bahwa warnanya lebih pudar begitu ya daripada mungkin pembanding begitu. Mungkin bisa dijelaskan struktur rambut itu apakah juga sama-sama polar sehingga bisa menempel dengan baik beta cyanidnya begitu. Mungkin itu pertanyaan dari saya, Bapak. Terima kasih.
Oke, can I terus Indonesia, Miss?
Yes, you will.
Baik, terima kasih Kak atas pertanyaannya. From the solubility of beta-cyanin is this is a solubility in water, yes. Why we choose the oil in water in preparation? Because preparation in oil in water is mudah untuk dibersihkan gitu loh. Mohon maaf. Terus
Easy to clean, yes.
easy to clean, yes, easy to clean. And so we make the leg system is to maintain the pigment. Yes. The next is mengapa tadi kok milihnya tetap itu ya? Alasan terbesarnya mungkin itu sih, Mbak, itu kalau acara saya. Terima kasih, Mbak.
Okay, thank you. Because the time is over, I will thanks for Mr. Sobari. Yeah, you make a presentation very good and give applause for Mr. Sobari. You can get back to your seat. Okay, thank you. Okay, next I will invite the next presenters, namely Galih Hidayanto. The floor is yours to make a presentation, please.
Okay. Thank you for Miss Ririn. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. I would like to say thank you for coming to my presentation. It's great to see you all. Let me introduce myself. My name is Galih Hidayanto. I am from Hasyim University. In this presentation, we will discuss application of leg system in formulation water in oil, hair dye, cream of butterfly pea, ethanol extract with twin span propylene glycol combination. Next. My first introduction, butterfly pea contains flavonoid in the form of purple anthocyanin, which have the potential as natural dyes. The anthocyanin content in butterfly pea reach 40.858 milligram liter. Mardiana et al. 2019. Next, leg system. Leg system. This is alcohol. Legs are a color pigment in the form of bond to a substrate in a complex system for through coordination bond. The leg system is used to increase the stability of color pigment in preparation from the influence of light. Next. Okay. Next, cream is a pharmaceutical preparation contain one or more will disparate medicinal ingredient in the form of water in oil or oil in water emulsion, containing not less than 60% water, intended for topical application to the skin. Cream preparation are widely chosen as topical preparation because they are easy to use, as good protection, comfortable, and easy to spread evenly to on the skin. Saran et al. 2013. Next, variety of emulsifier. The choice of emulsifier in the manufacture of cream is an important factor because the quality and stability of an emulsion is influenced by the emulsives used. Next, research. What are the physical characteristics of the preparation of hair coloring cream, water in oil, butterfly pea extract, ethanol with a combination of twin span propylene glycol? To second, how is the coloring ability of the cream of butterfly pea extract, ethanol with the combination of twin span propylene glycol? To next, I am to know the physical characteristic of the preparation of hair dye cream with butterfly ethanol extract with a combination of twin span propylene glycol. The second, to know the coloring ability of butterfly pea ethanol extract cream with a combination of twin span propylene glycol. Okay, next method. First, plant determination. Next, preparation of ethanol before I am pollination and maceration. And maceration to preparation of ethanol extract butterfly pea. I am phytochemical screening. Next, preparation of flag system. Hydrat alumina plus aerosil. We sieving this extract system lag. Next, preparation of hair dye pomade cream and evaluation physical, organoleptic, coloring, and homogeneity. Next, result and discuss. Butterfly pea ethanol extract yield was 29.1%. Phytochemical screening showed positive result for flavonoid, saponin, phenolic, and terpenoid. This indicates the possibility of anthocyanin pigment. Next, hair coloring cream, water in oil. Formulation 1, 2, and 3 show a blackish gray color, semi-solid with a characteristic odor. However, formulation 1 has a slightly rougher texture than formulation 2 and formulation 3. The homogeneity of formulation 1 is less than perfect compared to formulation 2 and formulation 3. Yes, result and discuss my analysis MANOVA 11 test significant more than 0.05. There is no difference. This is a reg picture, reg stable descriptive statistic. Next. This is a multivariate test. variation concentration, there is no difference because significant more than 0.05. This picture hair condition before and after treatment and observation for 120 minutes. Yes, next summary. Hair coloring cream, water in oil, ethanol extract of butterfly pea with variation concentration of twin span propylene glycol had good characteristic at each concentration. However, there is no significant effect on the physical characteristic of the preparation. Cream stick will to hair but then tend to fade more than cream color. Acknowledge the author would like to thanks profusely for the rich research funding assistance from LP2M, Dipa Wahid Hasyim, University Central Semarang. This is my reference. Thank you for your attention.
Okay, thank you for Mr. Galih Hidayanto, right? Next, any question from the audience? Can you stand up and mention your name?
My name is Shifa Maulida. Question is the reason why this formula is made use three different concentration of emulsifier.
Repeat to
Yes.
question.
The reason why this formula is made use three different concentration of emulsifier.
Yes, thank you for Siva. The cost of emulsifier in the manufacture of cream is an important factor because the quality and stability of an emulsion is influenced by the emulsifier. And surfactant and consurfactant as emulsifier. Twin and span as surfactant and propylene glycol consurfactant. Surfactant and consurfactant are very important ingredient in cream preparation because they are determine the role of emulsifier in the formulation of cream. Yes.
Thank you.
The time still enough. Next question from the audience. Are there questions? Okay.
My name is Tiara Tunima.
Yes.
Why is there no significant difference in the Monaco statistical test in the test of the three formulation? What is the ration? Yes.
Repeat to question.
What? Oh,
Repeat.
lagi. Why is there no significant difference in the Monaco statistical test in the test of the three formulation? What is the ration?
Statistic Manova, yes.
Yeah,
Oh,
Manova.
yes. It can be said that the rank of variance of the emulsifier in the study is not much different in terms of significance after being tested with Manova. The result are not significant or none. Yes.
Okay, the time still enough. Yeah. The next question, maybe other audience, please. Any question? Okay. If there are not questions, okay, please.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Saya mau bertanya tapi pakai bahasa Indonesia nggak apa-apa ya, Bu?
Nggak apa-apa. Ya.
Jawabnya pakai bahasa Indonesia juga nggak apa-apa. Saya mau bertanya, pada biasanya kalau misalkan pewarna dari bahan alam itu kan tidak bertahan lama ya. Terus apakah ekstrak bunga butterfly pea ini tadi dengan formulasi yang Mas jabarkan tadi, apakah dia bisa bertahan lama di rambut yang diwarnai gitu? Sedangkan yang kita mau pasaran biasanya kan pewarna rambut itu kualitasnya dilihat dari ketahanannya ya. Gitu aja. Terima kasih.
Kira-kira berarti tahan berapa lama gitu ya? Tadi, ya. In formulation cream bahasa Indonesia saja ya. Formula yang krim yang kami buat air dalam minyak. Saya bertujuan untuk agar tahan lama dengan apa? Dengan rambut dan itu kan pewarna alami nggak tahan lama gitu ya tadi. Tahan lama di sini ada sistem lag itu meningkatkan pigmen jadi sehingga apa? Tahan itu. Krimnya tidak mudah teroksidasi oleh air dan udara. Gimana?
Enough? Yes, because the time is over. I will mengucapkan selamat ya. Selamat kepada Mas Galih Hindayanto yang sudah mempresentasikan. Jadi next you can back to your seat. Thank you very much for your presentation and give applause for Mr. Galih Hindayanto. Thank you. Okay, next presenters. Ihsanur Laili Safara. The floor is yours to make a presentation.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Okay, good morning ladies and gentlemen. It is special for me to be here to present my research. First of all, let me introduce myself. My name is Ihsanur Safara and I'm from Hasyim University, Semarang. Thank you for the opportunity to present my paper entitled Antibacterial Activity of Ethanol-Methanol Extract of Mint Leaves or Menthapiperita L against Propionibacterium Bacterium acnes bacteria. Okay, this is my research background. Acne is skin disease that commonly affect adolescents and adults. Around 80 to 85% occurs between the age of 15 to 18 years old. And acne can be caused by a variety of bacteria, include Staphylococcus aureus, Staphylococcus epidermidis, and Propionibacterium acnes. And the secondary metabolite secondary secondary metabolite secondary found in mint leaves include flavonoid, phenolic, alkaloid, saponin, and tannin. And the sum of these secondary metabolites that can be used to antibacterial activity. And according to Farnat et al. 20 and 14, the combination of ethanol-methanol extract of mint leaves has a maximum flavonoid content of 3.33 gram quercetin per 100 gram. So the combination of ethanol and methanol is used to produce maximum flavonoid. Okay. This is my research question and the aim of my research. My research question, the first is, what are the active compound in the ethanol-methanol extract of mint leaves or Mentha piperita? And the second is there any antibacterial activity in the ethanol-methanol extract of mint leaves against Propionibacterium acnes? And the third is there any difference in the antibacterial activity of each concentration of ethanol-methanol extract of mint leaves against Propionibacterium acnes? And the aim of my study is to determine the presence of ethanol-methanol extract of mint leaves, and in addition to determine the presence or absent the active compound and presence of or absence of antibacterial activity and difference in antibacterial activity at its concentration of ethanol-methanol extract of mint leaves. Okay, this is my research method. The first is plan determination. Plan determination is carried out at the Diponegoro University. And the second is making of mint leaves simplicia into powder form. And the third is making ethanol-methanol extract of mint leaves using by maceration method and using the combination of ethanol 70% and methanol. And the fourth is phytochemical screening tests such as phenolic, flavonoid, alkaloid, saponin, and tannin. And the fifth is antibacterial activity test. The first thing to do is sterilization of tools and materials. The second is making Bernhardt infusion and Muller-Hinton agar media. And the third is preparation of McFarland standard solution. And the fourth is preparation of bacteria suspensi and preparation of the test solution, and then the antibacterial activity test by this diffusion method. And then the analyze the data with one-way ANOVA test. Okay, this is the research and discussion. The following are the phytochemical screening tests. The ethanol-methanol extract of mint leaves has revealed the presence of phenolic, saponin, and flavonoid. Because on the phenolic test, there has been a change in color from brownish yellow to blackish green. And the flavonoid test, there has been a change in color from brownish yellow to greenish yellow. And the saponin test is a foam was formed after desiccating it. And the ethanol-methanol extract of mint leaves negative to contain alkaloid and tannin because there is no precipitation after the test. Okay, this is the results of inhibition zone that forms around the paper disc. Based on this result, indicate a clear area is formed around the paper disc, and the higher the concentration, the higher the inhibition zone form. The table on above is results of inhibition zone diameter data, and the table on below is a picture of the inhibition zone that forms around the paper disc. Okay, this is the results of statistical data based on Sapir-Whorowicz and Lieben test. The result show that the significance value of more than 0.05, that the data is normally distributed and homogeneous, so it was continued with one-way ANOVA and post-hoc test. And based on one-way ANOVA and post-hoc test, the results show the significant difference at its concentration of ethanol-methanol extract of mint leaves. Okay, the summary. The first is based on phytochemical screening test, the ethanol-methanol extract of mint leaves has active compound such as phenolic, flavonoid, and saponin. And the ethanol-methanol extract of mint leaves has antibacterial activity against Propionibacterium acnes due to the formation of radical zone or a clear area around the paper disc. And then based on one-way ANOVA and post-hoc test, the results show that there was a significant difference of its concentration of ethanol, methanol extract of mint leaves. Okay, the acknowledgement. I would like to thanks to warm help in completing this research. Firstly, I would like to express my appreciation to Allah Subhanahu wa Ta'ala, and then to Miss Risa Filah Fitria and Miss Erika Indah Safitri who have provided guidance, advice, and motivation for completing this research. Finally, hearty and special thanks are due to my family for their appreciation and encouragement. This is my reference to completing this research. Okay, thank you very much for your attention. Maybe it's enough presentation from me. Thank you.
Okay, thank you, Miss Ihsanur Laili Safara. Okay, the audience, are there any question for the presenters related to the material presented by the presenters? Okay.
Okay, thank you. My name is Sari from Stifar Semarang. My question is, what's your reason to use the combination of the solvent, ethanol and methanol, to extract the active compound in mint leaves? And the second question is, you use 70% ethanol, then better than 96% ethanol. Why your reason to use that solvent? Maybe that's it. Thank you.
Okay, thank you for the question. I would like to answer the question. Why I use the combination of ethanol, 70 and methanol. Alright, okay. I am according to research of Farnat et al. 20 and 14 that the combination of ethanol and methanol in mint leaves, it has a maximum flavonoid contents. That flavonoid can be used to inhibit the bacteria, include Propionibacterium acnes. And then why I use the 70 lebih memilih 70% daripada 96 gitu. Because I use the polar solvent. Is enough?
Okay. When we use the 70% ethanol, is the polar than 96%, right? But why you combine with the methanol that it's non-polar? Actually, what is the main active compound maybe in the mint leaf? So you use the combination of two solvent maybe. Thank you.
Okay, I use the 70% and methanol because the active compound that yang akan saya gunakan, that I use is yang akan saya buktikan gitu, itu flavonoid, phenolic, tannins that is polar active compound. So I use the combination of ethanol 70% and methanol that's polar solvent. It's enough.
Enough, Mrs. Ungsari?
Yeah, thank you.
Thank you very much for Mrs. Ihsanur Laili. Okay, because the time is up, I hope you can stop and you will back to your seat. Okay?
Thank you.
And give applause for Mrs. Ihsanur Laili. Thank you very much. And the next, I invite Eva Monica, the next presenters, okay.
Thank you for the opportunity. Good morning, ladies and gentlemen. At this time, I want to deliver my presentation. My name is Eva Monica. I come from University, Malang. My presentation with the title is the use of cellulose extract from alang-alang Imprenta cylindrica L as filler and disintegrant of paracetamol tablet. Yeah. Pharmaceutical industry in Indonesia, as we know, has not been able to produce raw materials, medicine raw materials independently. Indonesia still relies on imported raw material. And as we know, medicine consists of raw material and active materials. And one of which is cellulose. Yeah. And actually, we can get this cellulose from Imprata cylindrica. This plant is suitable for commercial use because of its high cellulose content, short growth cycle, and it spread. Yeah, this plant is spread in various regions in large quantity. Therefore, the aim of this research is to find out whether cellulose obtained from Imprata cylindrica L is suitable to be used as filler and disintegrant of paracetamol tablet, and to obtain the optimal formula for cellulose from Imprata cylindrica leaf as filler and disintegrant. This is the workflow of my research. The first is Imprata cylindrica leaves were taken, washed and dried, and then the cellulose we obtained, the leaves we obtained were reduced in particle size, so we can get the powder fiber of cellulose, and then those purified cellulose powder will be tested for the characterization using the FTIR method, and then followed by the manufacture of paracetamol granule and tablet. This paracetamol granule and tablet following the three formula, with the difference in each formula is in its concentration of Imprata cylindrica cellulose and Avisal. We can see it here. Yeah. And then the paracetamol granule and tablet obtained, we carried out the process, and then we follow by the quality testing, characteristic quality testing, including this one. And then the data obtained were optimized using design expert software with a method in mixture simplex lattice design with 2 factors. This is the result. The Imprata cylindrica leaf powder that has been reducing in particle size, then with 3.5% nitric acid and sodium nitrate at 90 degrees Celsius, and then filter. The filter we use, then washed until neutral, and this process is repeated with using sodium hydroxide 2% and sodium sulfate 2%. And then the filtrate was bleaching using the sodium hypochlorite. And then the purification, we use sodium hydroxide and then dry. We can see here from the IR spectra, we confirm that this powder of cellulose we get were indeed really the cellulose, as comparison is standard pure cellulose, and the yield in this process is 16%. This is the result of the granule characteristic and tablet characteristic. The granule and tablet characteristic almost met the requirement, but not in the hardness and disintegration time. Yeah. This is mostly with the formula with the cellulose containing. And the particle distribution test also, the Avisal has not met the requirement, but this combination of cellulose and Avisal and cellulose itself, it met the requirement. And as we can see in the moisture content here, the formula with the high higher water content, it might be it might be from solid bridge, and then it causes the higher hardness and disintegration time. But the unique thing about this cellulose is, although it has high hardness and disintegration time, but the tablet is still fulfill the dissolution required because of its porous nature of the cellulose. It has the water of dissolution media can enter the matrix tablet with swelling mechanism. From the data, then we obtain the optimization of the formula in which the avisal pH 101 and Imperata cylindrica cellulose is 6.62% and 8.38% with the response is acceptable in the requirement. This is the summary. Summary of this research is cellulose obtained from Imperata cylindrica A assisted with avisal is suitable to be used as filler and disintegration in the manufacturing of tablet, but it is assisted with avisal. And the optimal formula for cellulose from Imperata cylindrica leaf as filler and disintegrant for a paracetamol tablet are 6.62% and 8.38%. And the future development have been searched, and there's lack of research that has been done to develop the micro and nanocrystalline cellulose isolation from Imperata cylindrica, and it can be used as control release agent, and then might be inspired to explore the cellulose producing potential of other Imperata species. And this research actually has so many weakness and limitation, one of which is the low yield value of cellulose extraction, and it can be optimized in the delignification process. And then less in-depth characterization. The characterization is still too simple, and it can be added with maybe XRD. And then the difficulty and other limitation is the voluminous bulk powder of cellulose made it difficult to process. And I thank to Machung University for the fund for this research in Machung Research Grant. And this is the library I use. Thank you for your attention.
Okay, thanks for your presentation, Ms. Eva Monica. Okay, the audience, there are any question? Any question? Please. Are there any question from you? Okay, nothing. If there are no question from the audience, I will ask you, Ms. Eva Monica, yes. In your research, why, what are your reason you choose cellulosa from Imperata cylindrica L? It as ingredient in tablet, right?
Yes.
Yes. What are your reason you choose it?
Yes. Thank you for the question. Why did I choose the cellulose? It's because cellulose, we use it almost in every tablet. Almost in every dosage form we have, we use cellulose, microcrystalline cellulose, and another cellulose. And we still import it until now. From several years ago until now, we still can produce the raw material itself. And Actually, the cellulose is we can get the cellulose from Imprata cylindrica. Alang-alang atau or Imprata cylindrica has 40% of cellulose content. Then I thought that it is very potential for the cellulose source.
Enough. Okay. Thank you for your answer, Eva Monica. Because the time is up. Give applause for Mrs. Eva Monica. Okay. You can back to your seat. The next presenter, I will be called Farah Fadia Anwar. The floor is yours.
All right. Thank you for moderator for the time. Assalamualaikum warahmatullahi wabarakatuh. Good morning, the honorable ladies and gentlemen. My name is Farah. I'm from Fakulty of Pharmacy, Wahid Hasyim University. And for this occasion, I would like to present my research and the title Anti-Aging Activity of Ethanol-Methanol Extract of Mint Leaves, Mentha piperita L on red. Okay. The anti-aging process is decrease of the collagen on the skin and weakening of the skin's barrier structure, which is this structure is caused by UV rays, especially UVB. Long-term exposure of UVB can form free radicals that decrease the elastin and collagen. And the previous study report that mint extract has an anti-aging activity by in vitro. And we use a combination with ethanol and methanol for the solvent to extraction for this research because the combination of solvent result that higher the flavonoid and anthocyanin contents, which is we know that flavonoid and anthocyanin play role as antioxidant. So we can use the combination with we use the combination solvent rather than its single solvent. And then from result of Sukahara et al. 2001 report that a plant extract was able to provide the anti-aging effect depending on the dose. So there are 2 points here for research question here. The first is, does the ethanol-methanol extract of mint leaves, Mentha piperita, have anti-aging activity on rats induced by UVB rays?
And the second is, does the increasing concentration of ethanol methanol extract of mint leaves, the greater the anti-aging effect it causes. And the purpose of this research is to determine whether the ethanol methanol extract of mint leaves has anti-aging activity on rats induced by UVB rays. And then knowing whether the increasing concentration of ethanol methanol extract of mint leaves, the greater the anti-aging effect. So let's move on to the method. First of all, mint leaf extract by mint leaf were extract by maceration method with a mixture of 70% ethanol and methanol in 1-to-1 ratio. And then rats were divided into 6 groups, each 2 rats each of the group, and 5 of the groups, which mean the treated group, were exposed to UVB, and followed by topical application of the ethanol methanol extract of mint leaves, or extract ethanol methanol down mint (EMDM). So we namely the groups with K2 for concentration 20 milligram per milliliters, K3 for concentration 40 milligram per milliliters, K4 for concentration 60 milligram per milliliters, and K5 for concentration 80 milligram per milliliters. And K6 is a negative control. We use the MSO. And then one of the group is also control group, but no exposed to UVB light and not use treated is as normal control. We call K1. And then we start rats induced by UVB with 311 nanometers with duration 20 minutes per day for 2 weeks. And then wrinkle, erythema, and exfoliation were scored on day 14 by the veterinarian. And this is Table 1 and Table 2 is assessment for the scoring to its parameters. For Table 1 is for wrinkles parameter. For Norof's wrinkles, we score 0. Slightly rough wrinkles, we score 1. Some rough wrinkles, we score 2. And some deep rough wrinkles, we score 3. And for Table 2, for scoring the erythema and exfoliation parameters, which mean for not any, we score 0, a few we score 1, mild we score 2, and many we score 3. And then this is the observation on day 14. For K1 is normal control and K6 for negative control. statistically, the score for wrinkles, erythema, and exfoliation in negative control is significantly different with normal control, and which means it can be said that UV rays were able to damage the skins of hind limbs of rats. So, the anti-aging activity was determined for wrinkles, erythema, and exfoliation, which is the smaller of the number of these parameters, the more effective the mean leaves of give the anti-aging activity. So this chart, we know that for K4 in wrinkles parameter is the smallest of the score, and for in erythema parameters, the smaller score is K2, K3, and K4, and for exfoliation parameter in K2 and K4, which the smallest of the score.
I'm so sorry, Miss. The time is up, so I will start to discussion.
Can
Okay.
I wait a minute until summary?
Okay, 1 minute, Ska.
Thank you. Okay, but statistically, there was no difference in the test group compared the negative control on its parameter. So even though there was a decrease on the mean score was smaller than the negative control, the difference was no significant. And the conclusion is ethanol-methanol extract of mint leaves at concentration 20, 14, 60, and 80 milligram per milliliters did not have anti-aging activity on rats induced by UVB light. And then the increasing concentration of ethanol-methanol extract of mint leaves had no effect on the anti-aging effect.
Okay, I'm sorry. I will stop your presentation. Okay, and then I will start for discussion. For the audience, any question? Okay, please stand up and mention your name.
Okay, thank you. Ms. Farah, I want to ask you, how much sample volume is used for topical application or your research? Thank you.
Okay, thank you for the question. Firstly, we oriented for the amount for the volume, but we have the literature which use 10 microliters for the volume. So after we oriented the amount of volume, that was sufficient. So in this research, we use 10 microliters for the volume of the application. That's it.
Okay, I'm so sorry for Miss Farah Fadia because time is up. So it must stop. Okay, you can back to your seat. Thank you for your presentation and give applause for Miss Tiaratun Farah Fadia Anwar. Okay, and next I will invite next presenters Tiaratun Nikmah. The floor is yours.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, nice to meet you and thank you very much for accepting for me. My name is Tiara Nikma. I am from the Bachelor of Pharmacy program at Hasyim University. Allow me to present my research with the title Molecular Docking Combination of Quercetin, Keveron, and Luteolin Compounds in Onions Again Main Protease of SARS-CoV-2 as anti-COVID. COVID is an infectious disease caused by the SARS-CoV-2 virus. By the end May 2022, more than 522 million people have been confirmed infected worldwide, with more than 6 million deaths. Anti-COVID therapeutic agents are urgently needed in the treatment. Main proteases play an important role in the viral life cycle of replication. Onions structure flavonoid compounds are known to have antiviral activity. The main components of onions are flavonoid compounds such as quercetin, keveron, and luteolin. Prediction of compounds activity on the selected target is done by in silico molecular docking. Leisure question. How is the interaction of the combination of quercetin, keveron, and luteolin compounds in onions with several amino acids and impro PDB code 6LU7, 3INS, 5RU7Y as an anti-COVID candidate based on molecular docking result? I am knowing the interaction of the combination of quercetin, keveron, and luteolin compounds in onions with several amino acids and impro PDB code 6LU7, 3INS, 5R7Y as an anti-COVID candidate based on in molecular docking. Method. The first is validation, which includes protein preparation and drug ligand preparation, redocking, and RMSD analysis. The second is the molecular docking test compound and comparison, which includes preparation of the validated protein and drug ligand, preparation of test the comparison compounds, docking of test and comparison compounds, and docking score analysis. The last visualization docking result with like Plus and Primal. Result in this question. The validation process aim to validate, aim to use provide that RMSD less than 2 Armstrong. PDB 6LU7 obtain value of RMSD 1.8873. PDB 5R7Y obtain RMSD value 1.5476. PDB 3A9S obtain RMSD value of 0.4473. The docking value is compound ligand native, obtain minus 131.45, combination quercetin, camphor and luteolin, minus 84.7115, hesperidin, minus 72.961, lopinavir, minus 124.446, Paxlovid, minus 99.33. And to PDB 5R7Y, obtain native ligand minus 64.0889, combination quercetin, camphor and luteolin, minus 73.393, IC39, hesperidin, minus 78.358, lopinavir, minus 108.022, Paxlovid, minus 88.6999. The list for PDB 3N9S obtain native ligand minus 111.404, combination quercetin, camphor and luteolin, minus 81.3577, hesperidin, minus 96.9488, lopinavir, minus 122.32.397, and Paxlovid, minus 105.684. The stability of the interaction of the compounds indirectly proportional to the potential of the compounds to give effect. The lower the docking score of the compounds, the more stable the complex bond between the receptor protein and the ligand. The most stable compound showcases at PDB6LU7 were native ligand, lopinavir, Paxlovid, combination quercetin, camphor and luteolin, and hesperidin. The PDB5R7Y are lopinavir, Paxlovid, combination quercetin, camphor and luteolin, hesperidin, and native ligand. Meanwhile, the PDB3A9S are lopinavir, native ligand, Paxlovid, hesperidin, and combination of quercetin, camphor and luteolin. Summary. Based on the result of molecular docking, the combination of quercetin, camphor and luteolin compounds from onions can interact with amino acid and improve SARS-CoV-2, PDB code 6LU7, 5R7Y, and 3A9S, so that it can be used a candidate model for anti-COVID, have a less potent effect when compared with lopinavir and Paxlovid. Future development or study. It is necessary to modify the structural design of the combination of quercetin, camphor and luteolin to obtain a more stable docking score to increase the binding interaction of the main protease. It is necessary to further research in vitro for the stable compound combination of quercetin, quercetin, and luteolin for onion LMCP. Acknowledgement, I would like to express my deepest appreciation and gratitude to my supervisor, Mrs. Anita Dwi Puspitasari and Mrs. Apita Gaisina Gaisani, the lecturer of the Fakulty of Pharmacy, Hasyim University, Semarang, Indonesia. In the reference, thank you.
Okay, thank you. For your presentation, Ms. Tiaratun Nikmah. Next, for the audience, any question? Okay.
Okay, thank you. My name is Sari from Semarang. My question is, in which functional group in camphorol or lutein in onion that you suspect can interact with protein in SARS-CoV-2? Okay, thank you.
I'm sorry, please.
Okay, sorry. In which functional group in the structure of camphorol or lutein in onion that you suspect can interact with the protein in SARS-CoV-2, and maybe how the mechanism that that functional group can maybe inhibit the protein growth in SARS-CoV-2. Thank you.
Which is a combination interaction.
Yeah, okay.
Choice luteolin and quercetin, but interaction, yeah. This is. I'm sorry.
Yeah.
Indonesia.
Oke, mungkin dalam bahasa Indonesia, Bu, sebenarnya di bagian gugus fungsi yang mana sih dari camphorol atau lutein begitu ya yang Anda suspek sebelumnya, yang Ibu perkirakan sebelumnya itu bisa berinteraksi begitu dengan protein sehingga penelitian ini dilakukan seperti itu. Terima kasih.
Baik, saya molekular daging ketika uji visualisasi itu terjadi interaksi beberapa gugus interaksi hidrogen dan hidrofob yang mana itu faktor terjadi pengikatan antara reseptor mpro dan tiga senyawa tersebut.
Oke, berarti mekanismenya bisa menghambat begitu ya, ketika dia bisa berikatan begitu. Cuman gugus fungsi yang mana belum diteliti lebih jauh, mungkin begitu ya, Bu. Ya, terima kasih.
Thank you. The time is still enough. So for audience, other audience, are there any question? Okay.
It was explained that the validation result must be less than 2. Why is that? Thank you.
Equation less than 2 because The smaller the RMSD value means the better, because the prediction ligand pose are getting close the conformation alternative ligand.
Okay, enough. All right, because the time is up, so thanks for your answer, Miss Tiaratun Nikmah, right? And you can back to your seat now. And please give applause for Miss Tiaratun Nikmah. Thank you very much. Okay, next I will be called next presenters, Shifa Maulida. The floor is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Diva Maulida. I am from Fakulty of Pharmacy, Wahid Hasyim University, Semarang. Here, I will present my thesis with the title Application of Lake System in Formulation Oil in Water High Day Cream of Butterfly P Clitoria ternatea L Ethanol Extract with Twin Span Glycerin Combination. Introduction. Butterfly P has a natural blue pigment anthocyanin type delpihidine glycoside rising 40.58 mg per liter. The anthocyanin found in the butterfly P have good stability in addition to the pharmacological potential of the butterfly P, among other, as an antioxidant, antibacterial, anti-inflammatory, and analgesic. Cream. Cream is semi-solid dosage form in the form an emulsion containing one or more medicinal ingredient dissolved or dispersed in suitable base material and containing not less than 60% water. There are two types of cream, namely the oil in water types and the water in oil types. The water-soluble oil in water cream is intended for cosmetic and aesthetic use. Variety of emulsifier. The choice of emulsifier in the manufacture of cream is an important factor because the quality and stability of an emulsion is influenced by the emulsifier used. The important ingredient in cream are surfactant and co-surfactant. In this study, the surfactant include twin and span, while the co-surfactant were glycerin. Lakes are a color pigment in the form of bond to a substrate in complex system formed through coordination bond. The lake system used to increase the stability of color pigment is preparation from the influence of lake, chemical factor, and heat. This system can change compounds that are initially water soluble to nonpolar. Research question and aim. What are the physical properties of the high day cream oil in water ethanol extract of butterfly P with a combination of twin span glycerin? How is the staining ability of the ethanol extract of the butterfly P cream with the combination of twin span glycerin? So now the physical properties of high day cream preparation of ethanol extract of twin span glycerin butterfly P extract. to determine the coloring ability of the ethanol extract of butterfly pea cream with a combination of twin span glycerin. Method. The first plan determination. Next, preparation of ethanol extract of butterfly pea. Before that, the first thing to do pollination and maceration. Continue with phytochemical screening. Next, preparation of lake system. By adding hydrate alumina plus aerosil, next shifting. Next, preparation of hair dye cream. Next, evaluation organoleptic, physical, and coloring. Result and discussion. Butterfly pea ethanol extract yields 29.1%. Phytochemical screening show positive result for flavonoid, phenolic, saponin, and terpenoid. This indicates the possibility of anthocyanin pigment. Hair coloring cream oil in water, ethanol extract of butterfly pea. F1, F2, and F3 show a blackish gray color, semi-solid with a characteristic odor. However, F3 has a slightly rockier texture than F1 and F2. The homogeneity of F3 is less than perfect compared to F1 and F2. Table number 1, next, the average value of each test can be seen in the table descriptive statistical. In the multivariate test table, the concentration variation does not affect the dispersion, viscosity, and pH. The significance is more than 0.05. In the Levin test table for the pH test, the dispersion power, adhesion, and viscosity have a significance of more than 0.05. So there is no difference. The following on the right is the condition of the hair before and after, and the left after treatment and observation for 120 minutes. Summary, hair coloring cream oil in water, ethanol extract of butterfly pea, with various concentration of twin span glycerin, has good characteristic at each concentration. However, there is no significant effect on the physical characteristic of the preparation. Cream stick well to hair but tend to fade more than cream colors. Acknowledge, the author would like to thank profusely for the research funding assistant for LP to MD Wahid Hasyim University, Central Semarang. This is my reference. Thank you for your attention.
Okay, thank you for your presentation, Ms. Shifa Maulida. Next, discussion. For the audience, are there any questions? Okay, please. Mention your name.
Let me introduce myself. My name is Nika Natania. So, Ms. Shifa, I want to ask you, you explained in the introduction that the butterfly pea flower contains anthocyanins, right? So I want to ask, Does butterfly pea flowers contain other compounds? Thank you.
Yes. Butterfly pea beside contain anthocyanin also contain flavonoid, glycosid, anthocyanin, flavonols, phenolic acid, terpenoid compound, and alkaloid, as well as cyclic peptide compound or cyclotide.
Okay. Next question. Okay, please.
My name is Farah. My question is why you choose the main ingredients of butterfly pea flower?
Because butterfly pea contain anthocyanin, flavonoid, and other ingredient, the anthocyanin content in butterfly pea reach 40.58 milligram per milli. The high level of anthocyanin from butterfly pea can be used as an ingredient for natural hair dye.
Okay, the time is up. Thank you for your answer, Miss Shifa Maulida. You can go back to your seat now and give applause for Miss Shifa Maulida. Thank you. The next presenters, Farida Maira Lokana. Okay.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Thank you for a good opportunity for me. Thank you for moderator for time for me. Allow me to introduce myself. My name is Farida Maira Lokana from undergraduate Fakulty of Pharmacy, Wahid Hasyim University Semarang, supervisor by Mrs. Apoteker Agnes Budiarti, Magister of Science, and Mr. Apoteker Khairul Anwar, Magister of Pharmacy. On this occasion, I'd like to present about research about entitled Antioxidant Activity of Deodorant Leaf Eugenia uniflora L ethanol extract and determination of total flavonoid and phenolic content. Next slide. There are something that discussed today, and then introduction. The picture showed that there are free radicals. Free radicals are more small molecule that have one or more free electron in outer shell, so they unstable and very reactive. They will react with surrounding molecules to get electron pairs and obtain of and achieve molecule stability. Next picture is condition oxidative stress. This is a normal shell, and in excessive amount, the free radical can attack shell. The normal shell is if occurs continuously, we can it can damage of shell disease. So it trigger of the degenerative or chronic disease. For example, cancer disease. So we need to something that inhibit of free radical. It's called antioxidant. The mechanism is antioxidant will give an electron to pairs a molecule of free radical. Next, antioxidant we can obtain from Dewandaru plant, Eugenia uniflora L, and this is several previous studies about Dewandaru leaves. Based of Santoso et al. 2020, Dewandaru leaves has a potential natural antioxidant, and then Dewandaru leaf contains a phenolic compound, alkylic compound, phthalic acid, and rutin by HPLC in aqueous ethanol and methanol acetone extract. And then isolate polar fraction ethanol extract Dewandaru leaves contain isoflavone. And then the fraction of ethyl acetate extract and ethanol extract of Dewandaru has high anti-radical activity. The high anti-radical activity was indicated by the ARP value of 3,496.5 milligram dppH per milligram sample, while vitamin A was 1,776.20 milligram dppH per milligram sample. Next research question and the aim. The question are: point 1, is there any antioxidant activity in the ethanol extract of Dewandaru leaves with ARP value using ABTS method? And how much content of total flavonoid and phenolic compound in the ethanol extract of Dewandaru leaves? The aim, this study aims to test the antioxidant activity of ethanol extract of Dewandaru leaves using the ABTS method, and study also determine the content of flavonoid and phenolic compound in total ethanol extract of Dewandaru leaves. There are the methods. First step, we do plant determination at Diponegoro University, and then making Dewandaru leaf extraction with the several process and phytochemical screening and TLC, and then ABTS method antioxidant activity test, and the last determination total flavonoid and phenolic content. Next slide, please. Result and discussion, material collection. Dewandaru leaf were collected from Susuan village, is Ungaran district, Semarang Regency, and then plan determination aims to ensure the plants used in the study so that error in plant selection can be avoided. And the result of the determination plan is the Dewandaru plan the result is the true that use is Dewandaru plan. This is making Simplicia powder and extraction by maceration. Maceration with using 96% ethanol solvent, and rendement of the extract obtained was 31.65%. And phytochemical screening, the ethanol extract hewan daru leaf contain saponin, flavonoid, phenolic compound, and tannin. And GLC as confirmation test, the result that the extract positively contain flavonoid and phenolic. Antioxidant activity, the result showed that the ARP value obtained from the IC15, and then we count ARP value from that. The result is 70.61 milligram APTS per milligram sample. This means that the higher ARP value, the more effective the antioxidant activity. And determination of total flavonoid, the results show that the ethanol extract hewan daru leaf had a phenolic content of 226 milligram gallic acid per milligram sample, and then determination of total flavonoid is showed that 35.65 milligram quercetin per milligram sample. This is a table of data analysis antioxidant activity, and then this is standard curve. This is analysis data of phenolic content.
I'm sorry, remind the time, 1 minute again.
Thank you. This is flavonoid content. In summary, the ethanol extract of the daru leaves has antioxidant activity by ABTS method. The ARP value is 70.61 milligram APTS per milligram sample. The higher ARP value, the more effective the antioxidant activity. And two, the ethanol extract of the daru leaves contain phenolic and flavonoid compound with level of 226.48 milligram gallic acid per gram sample, and 35.60 milligram quercetin per gram sample. Development study, it is necessary to test antioxidant activity hewan daru leaf with the same or different method with various extraction process, and determine the level of other compounds that have the potential as antioxidant. Weakness and limitation, hewan daru plants tend to be rare to find, and not many people know about them. And
I'm sorry, I must stop your presentation.
Okay, thank you.
Okay. Next discussion session for the audience. Any question? One question only. Okay. Maybe behind present. Okay.
Tes. Okay.
Mention your name, please.
Okay. My name is Isanur Laili Safara, and I want to ask to Miss Farida. My question is, can you explain how to get the ARP value? And is not it enough to determine antioxidant activity with the IC50 value? Thank you.
Okay, thank you. Thank you for the question. Okay. Here I want to show you how to get the ARP value, but I think it is not enough to get antioxidant activity from IC15. So, but we can analyze more to get a ARP value. Please show the slide, Mr. So, table of result. Naik, Pak. Tabel itu, tabel data. Yeah, yes. And this is a table data of analysis antioxidant. From measure of spectrophotometer, we can get absorbency, data of absorbency APTSN sample. So we can count percent inhibition. And then you can make standard curve between concentration of sample with percent inhibition. And then you can count IC15. It is not enough. So we can count continuously. IC15 can change from ppm to milligram per milli. So we can count SA15. SA15 is a concentrate which concentrates required to scavenge or inhibit of radical ABTS. To express milligram sample per milligram ABTS, per milligram per milli ABTS. So we can count ARP value from this. You can obtain ARP value for the formula by formula 10 per AC15. ARP value is described of the strength of anti-radical, where the height of ARP value, it means the height of antioxidant activity. Here, something like that.
Okay, enough.
Thank you.
Yes. All right. Thank you for your presentation. And next, you can go back to your seat and give applause for Miss Farida Meira Lokana. Thank you. And next presenters is Yancha Anas. The floor is yours.
Cek. Thank you, Bu Ririn, for the opportunity. Assalamualaikum warahmatullahi wabarakatuh. Yes, thank you very much for the organizing committee, Whicaps 2018, for allowing me to represent my supervisor, Ratna Asma Susidarti, Nunung Yuniarti, and Roni Martin, to present some of our research results with the title Development of Pentagamma Phenol Synthesis, Purification Improvement, and Crystal Isolation from Dorine Solvents. The background of our research is that pentagamma phenol, or more popularly we know as PG-phenol, slide please, is a curcumin analog compound with a broad pharmacological action. PG-phenol has a broad effect as antioxidant, free radical scavenger, anti-inflammatory, anti-cancer, antibacterial, antiviral, hepatoprotective, and anti-cholesterol. However, the previous author did not mention the purity of PG-phenol used, so the acting pharmacology is still debatable. Therefore, the purity data is we need in a very compound synthesis, and we expect the purity data is has a high purity. So, slide please. Several previous researchers has conducted the development of synthesis of PG-phenol. Lastly, Red Maleni in 2016, she stated that she shortened the reaction of PG-phenol synthesis to 2 hours. Because the reaction is needs time for 2 days until 14 days. she used the ethanol and hot water or aqueous to the rinse process. And hot ethanol is used for the phenol recrystallization. However, unfortunately, there is no information about the phenol purity. Using phenol in the rinsing process has the potential to dissolve phenol and allowing it to pass during filtration process and precipitate in the rinse solvent, like this picture. In addition, we predicted the phenol reaction still take a place after 2 hours of reaction. And therefore, we estimate that many phenol precipitate in the rinse solvent. As a frequency, we need the method to isolate phenol for the rinse solvent. Slide, please. The aim of this study is development of purification method, especially in the rinsing procedure, to attain a phenol with high level purity. And the second aim is to establish the method for purified phenol from the rinse solvent. Slide, please. In our work, we used the method or procedure developed by Virit Malini in 2016, and we react vanillin prosynthesized with cyclopentanone in round button flask with hydrochloric acid as a catalyst. The reaction took place in 75 degrees Celsius for 2 hours, and we use acetic acid and water or aqueous 1:1 ratio to extract the phenol. We separate phenol and rinse the solid or crude phenol with ethanol and boiling aqueous. The improvement is here. We rinse for 3 cycles and drain phenol in oven in 76 degrees Celsius for 3 hours and recrystallize with the 2 solvent system with hot ethanol and cold distilled water. For isolation phenol from the rinse solvent, we separate phenol with the liquid and extract phenol with acetic acid and water 1:1 ratio and rinse crude phenol with ethanol and boiling aqueous for 3 cycles. And then we dry in oven at 76 degrees for 3 hours and recrystallization with hot ethanol and cold aqueous. For characterization, we see the differences of synthesized phenol and isolated phenol organoleptically and randomly. Edge and number spectral data, we use edge and number spectral data to identify phenol chemical structure. And for fluidity analysis of phenol, we use 3 methods, especially melting point and melting distance, thin layer chromatography profile, and purity based on high performance of liquid chromatography. Next, please. What we got? We synthesized phenol for 10 repetition, and we got the 9.49 gram of phenol with 10.79% yield. And we isolate 3.16 gram of phenol from the rinse solvent with the rendement of 3.59%. There were, organoleptically, there were a slight difference synthesis phenol and isolate phenol. Synthesized phenol had a bright yellow color with a smooth appearance and did the adherence to filter paper and or glass vial. Meanwhile, the isolated phenol had an orange crystal color and adhere to filter paper and glass container. The picture one is HNMR data for synthesized PG-phenol. This spectral data identically with the previous author reported and a slight difference for hydroxyl group peak. In our study, hydroxyl peak appeared at chemical shift 7.183. Sarjiman et al. In 1997 reported this peak appeared at 8.6, and Dai in 2006 reported that hydroxyl peak appeared at chemical shift of 9.3. Therefore, this study confirmed because in general, the hydroxy attached to aromatic ring can appear in chemical shift between 4.5 until 7.2. Therefore, we conclude that that hydroxyl peak in the 7.183 is the hydroxyl peak attached to two aromatic ring in PG-phenol chemical structure. The second pictures, next, slide please, next. In the second picture, this is show us the TLC profile comparison of synthesized PG-phenol and isolated PG-phenol from the rinse solvent under 254 nanometer UV light. We can see that the two PG-phenol TLC profile look similar with single spot for PG-phenol in the right, and there is no spot for vanillin as starting material or impurities on our result. Based on melting examination result, the synthesized PG-phenol has a narrower melting distance, 1.3 degrees Celsius. Meanwhile, the PG-phenol in rinse solvent had a wider melting distance. From this finding, we conclude that the isolated PG-phenol had a lower purity than synthesized PG-phenol. For analysis of purity of PG-phenol, then we use the HPLC method with a UV light detector. Based on the scanning of lambda max, the PG-phenol had a maximum absorption in 219 nanometers. Meanwhile, we use the vanillin as impurity because the vanillin is starting material in a PG-phenol synthesis that may be still left in the final product. The PG-phenol, based on the spectra, PG-phenol appeared at a retention time for 4.2 minutes. Meanwhile, the vanillin appeared more quickly in 1.2 minutes. PG-phenol 100 milligram per milliliter produce a quick broad peak. Unfortunately, in this spectra, vanillin peak also appear in 1.2. After calculating the purity analysis of PG-phenol, the level of purity of synthesized PG-phenol was 96.9%. At the same concentration, we test the isolate PG-phenol and calculate the purity. We found the purity of isolate PG-phenol is 84.6%. Based on this data, we can conclude that the purification process of PG-phenol, especially in PG-phenol in the rinse solvent, is not optimal and require further research. Next, please. Next. Next. The conclusion of our research is the purification technique used for phenol synthesis was improved, the resulting in phenol with a high purity grade. Work also provide a method for isolating phenol in the rinse solvent. What the implication of our research? The future research or student like me can apply the rinse and recrystallization method to produce high purity phenol with consistent physicochemical properties. And the next scientist can optimize the phenol isolation technique for the rinse solvent to improve the synthesis product yield. Next. In this moment, I want to say thank to Universitas Hasyim because to supporting me in my PhD program. I will thank to BP Beasiswa Pendidikan Indonesia, Kemenristek Dikbud, and LPDP for doctoral program scholarship. And special thanks to Dr. Limat Leni, Professor Dr. Sarjiman, Faruk Parahmadinata, and Panji Pranata, UGM staff for their guidance and assistance for the phenol synthesis development and purity assay of phenol with HPLC and detifying structure with phenol with HNMR. Finally, thank you for listening and your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for your presentation, Mr. Yanja Anas. And next session for the audience, are there any question for Mr. Yanja Anas? Please. Okay, thank you, Mrs. Ungsari. Yeah.
Okay, thank you. My name is Sari Mr. Yanja. My question is, when we identified phenol with NMR, what can we do to be sure that it is a phenol? As we know, there is a PGV1 that has a similar structure with phenol. Okay, thank you.
Thank you for the question. It's a good point here. What we can do to differentiation of phenol and PGV1? Yes, first we can differentiate the phenol and PGV1 from the lambda max for UV scanning. And two, we can use the NMR data, because the NMR data phenol and PGV1 is very, very different, especially in hydroxyl peak that I show in this picture. Thank you.
How about the answer from Mrs. Yanja Anas? Do you
Yes,
understand?
it's clear. Yes, thank you, Mr. Yanja.
Okay, thank you, Mr. Yanja Anas,
Thank you
because the time is up, so you can go back to your seat. Thank you. Give applause for Mr. Yanja Anas. Okay. Okay, all participants, because all presenters has finished their presentation, so I will close oral presentation today. I'm so sorry for making mistake, and thank you for your participation. Goodbye, see you. Wassalamualaikum warahmatullahi wabarakatuh. Kepada seluruh peserta bahwa dipersilahkan Anda semua untuk isoma terlebih dahulu sampai dengan pukul 13.00. Silahkan Bapak Ibu semuanya, jika Bapak Ibu ingin istirahat sejenak dan makan, sholat, dipersilahkan.
adalah pemaparan materi dengan judul Nano-Based Value Care Solutions for Oncology Practices with Focus on Herbal and Nutraceutical yang akan disampaikan oleh Profesor Yashwat Patak, Doctor of Philosophy. Sesi ini akan dipandu oleh moderator Ibu Apoteker Ayu Sabrina, Magister Farmasi. Kepada Ibu Apoteker Ayu Sabrina, Magister Farmasi, disilakan.
All of the participants, the next session is presentation with the theme Nano-Based Value Care Solution for Oncology Practices with Focus on Herbal and Nutraceuticals by Professor Yashwat Patak, Doctor of Philosophy. This session will be moderated by Miss Apoteker Ayu Sabrina, Magister Farmasi. To Miss Apoteker Ayu Sabrina, Magister Farmasi, the floor is yours.
All right, ladies and gentlemen, can we begin the All right, let me begin. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, and dear ladies and gentlemen. Today, are you still excited for today's seminar? Okay, please pay attention for Professor Patak. Okay, all right. Today, I'm Ai Sabrina as your moderator today, and I'm very pleased to see you again, Prof. We meet on past. And today's seminar is about nano-based value care solution for oncology practices with focus on herbal and nutraceutical. And talking about cancer, it reminds me about one of our lecturers, the late Elia Zul, passed away during her fight against colorectal cancer. May she rest in peace and put in. Our speaker today, Professor Yashwat, he has over 50 years experience in institution of higher education and as a dean of the University of South Florida, and he is focusing on nanotechnology, drug delivery system, and nutraceutical. And he has traveled over 80 countries and gave talks in many universities and offered workshop and shared session and was a keynote speaker at many national and international conferences. So right now, before the presentation begin, let me inform you how the presentation will be going on. First, Professor Patak will invite to present the material, and after that, there will be a question and answer session, followed by the conclusion. Okay, Mr. Patak, the floor is yours.
Perfect. Selamat siang. Siang or malang. The food is so good that most of the people have eaten a lot. So malam will be more appropriate because it's sleeping time. So selamat siang. My talk is going to be nano-based value care solution for oncology practices with focus on herbal and nutraceuticals. I always show this slide. I am from originally from India, but we are in Tampa, Florida. This is the beautiful view of Tampa, Florida, and I welcome you all to visit us, and I will be able to take you all around. Terima kasih. Thank you very much. My special thanks to Ibu Risha and all the committee members of the VPAC 2022, Honorable Chair of this session, Ayush Shavarina. Am I correctly saying that? Fantastic. I got A+ now. And then all students attending talks, I see very few of them there. So special thanks to those who are sitting at the back and sleeping. After the good meals, you can do that. I will try my level best to make it slow and not very loud so that I will not disturb your things. Apa khabar? Mohon maaf, saya tidak bisa bicara bahasa Indonesia. Selamat to be apotheker. Selamat belajar. Keep calm and speak bahasa. You know, first time when I landed in Jakarta, it took me more than 3 hours in the taxi to reach Sudaraman. And the taxi driver, he didn't know a single word of English. I didn't know a single word of Bahasa. But then we talked about Shah Rukh Khan. And then he was knowing everybody. He was knowing Shah Rukh Khan, Salman Khan, Amir Khan, Aishwarya Rai and everything. We loved talking each other in Bahasa. He talked in Bahasa, I talked in Hindi, and we learned. So it helps a lot. So, nama saya Yashwant Pathak, saya berasal dari Tampa, Florida, Muluk, India. Saya bekerja University of South Florida College of Pharmacy. Saya Professor Associate Dean for Faculty Affairs. Ada pertanyaan, any questions? No answers now, because only 45 minutes. I was supposed to give talk for 2 hours, and then I was thinking that I will go in Kereta Api. Now my time has been reduced to only 35 minutes, so I am going in Japanese bullet train. Sounds good? So if you suppose cannot keep up with my speed, don't worry. Ibu Risha has got my PowerPoint presentation. Just send her a WhatsApp text and she will send you the whole presentation and you will not miss anything. Please continue your sleep now. So I have 3 points I am going to talk about. One is value-based oncology care. Second is nanomedicine in cancer treatment, recent developments and future trends. And third is herbal and nutraceutical for value-based oncology. I am very happy there is a one paper here where it is talking, a poster which talks about use of herbal drugs to reduce the anti-cancer activity and complement the activity. I was very impressed with a lot of these posters. Very nice posters have been done. And slight modification of adding nanotechnology, these posters will be of world class. Just have to slightly move from micro to nano, and it will be wonderful to all of you to do that. So oncology, everybody knows that, so I am not going to say too much of it, but oncology cost is significantly higher. It is too much. And oncology, we all know where it starts and where it ends, we don't know. Because it's a dysfunctional cells which create a lot of challenges in the body, and finally you start doing it. Some of the things which we now know is the heredity, like breast cancer is hereditary. Most of the people whose mothers have breast cancer, there is a lot of chance of they having breast cancer. Like that, there is a heredity component is there. There is another possibility is radiation. If you sit in the beach, like we have beautiful beaches, and if you sit in the radiation, you may get it. Then chemical, we are using a lot of fertilizers, insecticides. So there are many reasons which now science has developed to understand more and more about the cancer. And that is where you will find, but the cost is exorbitant. The amount of money which is needed to treat cancer is growing exponential. Lot of money is going. So you can see in 2015, the cost of cancer treatment in America was 182.6 billion. By 2030, it is going to be 45.6 billion. It is a lot of money which is needed for treatment of the cancer. But there is another positive thing, that the cancer survival rate is growing. So people in good old days, I remember 20 years back, if somebody will tell me that, oh, I have a cancer, I immediately say, oh my God, he is on way to Makam. Probably tomorrow, day after tomorrow, or a month. But now the things have changed. People are surviving for a longer time. I have a friend who is a doctor. Her husband died with pancreas cancer in less than 3 months. The wife also suffered from pancreas cancer. She is still alive after 5 years, just because she got the nanomedicine treatment for pancreas cancer. And her cancer was early detected. And this is where the key is there for the treatment of. So 2019 survey has said that Mesothelioma Center for Asbestos, they said that 63% of the cancer patients and loved ones suffered from financial challenges. And this is major part for the financial challenges which has been seen, because the cost is prohibitive. And you will find that people who have high income, their survival rate is 90%. People who have low income, their survival rate is 65%. So based on the amount of money you are making every month, your livelihood will be possible if you have lot of money. If you do not have a lot of money, you may die early. This is how this cancer cost is causing a challenge for the. And then there is another challenge in America is underinsured. If you have not full insurance coverage, then it is very difficult to treat the cancer, and it is very tough. This is the cancer in Indonesia. You all know this number, so you must have seen earlier. But what I want to point it out is that pharmacists can play a significant role to reduce the number of cancer deaths. One of the reasons is that if you can identify the cancer at the early stage, then the survival rate is higher. And for example, because we have many female audience here, they normally have uterine bleeding, uterine infection and then bleeding. And if that continues for 6 months to a year, and if you neglect it, then you come to know that you have a kidney cancer. Now, if you would have tested right in the beginning when you have UTI or uterine infections, you may know that you are having cancer. And then you be able to treat the cancer in the early stages. Same thing happens with breast cancer, with prostate cancer, with many different types of cancers. Early detection is the key for survival, and that's where the pharmacist can help, because pharmacist can tell them that, oh, this is problem, this is, you find it out, do CT scan or do PET scan, and you'll be able to get it. So there is a income disparity, and cancer, so higher the income, higher the survival, lower the income, because the treatment is not provided. And that is not only happening in small countries, but it is happening in economically developed countries. Even in America, it is a big problem there. So America, cancer is considered to be epidemic. We just came out of pandemic. I was wearing my mask, and our chair permitted me to not use mask, so I am not using it. But the US cancer is considered as epidemic, and almost 1,806,000 people are new cancer cases happened in 2020. You can imagine. And you can now understand the reasons also. And I want to congratulate you all, Indonesians, for your successful management of COVID. And you should pat your back, because in the world, Indonesia and many other smaller countries have made records as compared to America. We had 300 million people and more than 1 million people died. In my area in Florida, we have a senior citizens home where everybody from that home, 150 people died within 2 weeks, because they don't understand. And then they used the ventilators, and that's where they made a mistake, and rest of the world understood that oxygen level helps other than the ventilator. That was the difference. But I must congratulate the Indonesians for doing fantastic job in. So 606 people died in 2020, and in pandemic 1 million have died. So cancer is almost like pandemic. Number of people who are dying, but we don't give that much importance what we have given to COVID, because Pfizer was involved. And Pfizer made more than $50 billion in that COVID vaccine thing. So anyway, so cancer is like an epidemic in the things, and the number of possible cancer death rises dropped from 27% from 2001 to 2020, just because of the new treatments which have been happening. And those treatments are based on nanotechnology, new and newer nano. I am going to share you some of the things, but the cost is going up and up, and people are also, so it has become now second leading cause after the cardiovascular diseases in United States. And then second thing is post-operative cost. It is very exorbitant, very high. And that is why people don't go for it, because you don't want to go under surgery. So they prefer to die rather than going through surgery, because the post-operative cost is very high. And these are some of the data. I will leave it with you when you see these things. But everywhere you will find that the number of, look at this lung cancer. The annual cost is around $282,000. If you take the breast cancer, $150,000, $101,000, $165,000. So for different cancers, the amount of money which is required is different, and it is becoming more and more expensive. And why more people are getting cancer? Because more people are surviving longer time. Cancer is a deformity in the cells. As you live longer, the chances of having that deformity or cell mutation happens over the period of time. And as you age, the possibility of getting cancer is very high, because in 1960, the average American age was 70 years. In 2018, it is 81 years. And believe it or not, we don't know what to do with the old people. They keep on living and living and living. And that is where the challenges are coming, because they raise the cost of treatment, and that is the problem which is going on there. So this is the cancer research data from UK, where they are saying people were thinking that smoking will cause, but it appears that the smoking and non-smoking both meet at some point. So it is not only one reason, but there must be several other reasons in the cancer. So value of cancer care is growing significantly. We have seen that. And income disparity is creating problems for that. So if your average monthly income is around $3,600 and your chemotherapy cost is around $1,000 to $12,000 and radiation cost is around $9,000, where from you will bring the money? And that's where poor people die of cancer very quickly as compared to the richer people, and that is what is happening. So how nanomedicine is helping? So how nanomedicine in cancer treatment is helping, and we'll see some of the recent developments for the nanomedicine as well as future trends. So there are many different ways you are doing a delivery system of suitability of specific tumor pathophysiology, target population select criteria, what will be enable earlier top decisions, understanding therapeutic. What is happening is that whole cancer treatment is now moving towards personalized treatment. And you will find that all people don't need the same amount of drug. Now what happens is, you go to the pharmacy, you take 300 milligram for headache, whether you are an Asian or African or a 6 feet American bully, all take 300 milligrams. That's not good. So, and when it comes to aspirin, this is okay, but when it comes to the potent drug like cancer, anti-cancer drugs, that is not good. So people are now realizing that you cannot put the same amount of anti-cancer drug in all the bodies. They change, because some people are very, like I am short. That's why I was planning to talk from there, but then I realized you won't see me. So I said I will stand here, so at least you can see me. And most of the Indonesians are same height, so not to worry much. But so it is all depends upon personalized medicine. So cancer treatment is more and more becoming personalized treatment, and that is where the things are coming. And that's where I want to quote this, because whenever I quote this in any big conference, they criticize me very badly, because I say that when you start using statistics, you stop doing science. Because when you use statistics, you are trying to, in pharmacokinetics, I have worked in pharmacokinetics, there are 9 different models. If one doesn't fit, you go to the second. Second doesn't fit, you go to the third. And that is where the problem comes. So statistics, I want to repeat this, and this is, Mark Twain is a very well-known person. He said that there are lies, damn lies, and finally there is statistics. So take it with a pinch of salt, but that is fact of life here. So, if you see the different types of cancer cases in, they are growing because aging population is growing, and you will find myeloma, lymphoma, leukemia, all different types of cases, and it amount to around 172,000 cases or 173,000 cases. So cancer is and you will find that the survival in case of myeloma has changed now. Survival has become 50% almost. In case of Hodgkin's lymphoma, it is 89%, 73%, 63%. And from 1960-63 to 2006 to 2012, you will find that in last 30 years, because of the science, the survival rate have grown, and that is the advantage of some of the new technology which have been developed. So nano cancer is a nano medicine is one of the interesting things. So they are trying different types of early detection, diagnosis, most important. As soon as you find it, it is very nice to treat properly. So every year, if you have some of your biomarker tested, that will help a lot. And like I get myself tested for prostate cancer, and my doctor always tells, for coming 10 years, you will not get prostate cancer. That's what he tells. But God has different plans. I don't know what are those. So you have several nanomedicine products which are approved for clinical trials. So you have Myosate, Dexil, Thermodox. So these are all in different types of clinical trials, and they are all nanomedicines. Now you can have nanomedicine targeted to go to a specific tumor. And that's where you will find that 15 years back, when people used to have cancer, they will lose their hair, they will lose their weight, and lot of side effects of the anti-cancer drugs were there. Nowadays, they are not there. Because of the nanomedicine, you are reducing the amount of dose. You know, almost 10 times less dose is provided. So obviously, you see 10 times less side effects of the thing. And these are some of the things. This is another area, cisplatin, paclitaxel. All these drugs are given in different types of cancers: ovarian cancer, non-small lung cancer, malignant mesothelioma, gastrointestinal adenocarcinoma. You know, all these cancers, they are using it, and they are successful. That's what I told my friend's wife, still surviving after 6 years with pancreatic cancer because of this nano treatment. These are some of the nano treatments, nanomedicines available and approved by the FDA. And as I mentioned, they use different types of polymeric nanomaterial, polymeric missiles. Liposomes are very commonly used in the system. So there are future trends. Now what is happening is, in early days, there used to be only two alternatives. One is chemotherapy, other is surgery. And that's where most of the people have died early in cancer because of either surgery or because of chemotherapy, because of the side effects of the anti-cancer drug. But the things are changing now, and newer approaches are coming. Thought process is different, and now people are thinking about systems biology. And system biology approach is a very interesting thing, and I would like to tell you that our body is nothing but it is a network of the networks. So you have organ networks, then your cellular network, molecular network, and then your gene network. Now, this gene is responsible for certain types of cancer. And now the immunotherapy, the new technology which is developing, is addressing the cancer at the gene level. And because of that gene level, you don't disturb the rest of the networking. You don't disturb the networking of organ, networking of molecules or cellular, but you straight away go to the gene. So I have a friend who is also professor in Johns Hopkins. He used to be my colleague. He suffered from leukemia. He is still surviving for last 7-8 years because of the immunotherapy. And he has to take that immunotherapy now once in a year. They modify these genes which are existing or which may cause, they can clip the genes. And very interesting immunotherapy is being developed in cancer so that you don't see or you don't disturb the other networks. And this is a typical system biology approach which is going on. And then there are signaling pathways. We all know that most of these genes, they provide certain signals. And you can create those signaling pathways. People have done a lot of research in those signaling pathways. And now you can focus on a specific pathway which can be treated to prevent the growth of the tumor. or which can reduce the growth of the tumor. And this is where these signaling pathways are becoming part of the system biology treatment for cancer. So incidence of cancer is also correlated with aging. As I mentioned, as people age, the chances of getting the mutations is very high, and it is related to the aging process. And you will have suppose if you are 70 years old body, then you have almost, the genes might be mutating starting from 30 years. They have 40 years to mutate and completely convert it into a cancer. And this is why the aging population suffers from the cancer a lot. So challenges are, we are trying to understand this patient's biology now, and nanomedicine behavior, how it happens into the body system, and then which medicine can be applicable to a particular signaling pathway, or you can have combination of medicine given in nanomedicine format, so that nanomedicine can really help in combining the products and then reducing the effects of the side effects of the anti-cancer drugs. And this is where nanomedicine is significantly helping. And they are trying to have pre-selecting the patients likely to respond to the nanomedicine based therapy. So if you have 100 patients, then based on their genetic combination and all those studies which they do, biomarker studies, then they select it, which drug will be useful to this particular group of patients, which drug will be useful to that particular group of, and then now they are creating nanomedicine to treat that. And this is where you don't use the same medicine to all the 100 patients, but you select a group where this drug will be most effective in studying their patient biology. So there are many different technologies or patterns they are using. So understand the tumor type, specific pathophysiology, implement patient-focused design of new nanomedicine, bridge the gap between research and treatment, and employ patient selection and strategies. And here, this is the strategy which can come under the personalized medicine. And personalized medicines will help to reduce the side effects, will help to at least reduce the growth of the cancer cells and prevent from the immediate death. And this is where these personalized medicines can be used for using nanotechnology. So you have different types of ways. So you have a passive tumor targeting. You can target the passive tumor. It's a systemic. Put up an injection, it goes into the body and finally reaches there. Or you can have a active tumor targeting. What you do is you take the medicine, put it into liposomes or dendrimers, add some antibodies which can take straight away to the tumor. Now your other networking of the organs is not disturbed, so you don't get side effect. The drug can be delivered right into the tumor with the help of antibodies and the nanomedicine. And this is the technology which is going to be almost applied all over the world very soon, probably. It's like TV. When you bought the TV first time, it used to be very expensive. Now you go to the shop and pay $100 and you get the TV, because TV has become cheap. And that's where the same thing will happen with immunotherapy and nanomedicine therapy, that in due course of time, all these therapies which are so-called advanced therapies will be readily available to common man in every country, so that the lifespan will be increased and cancer can be treated. And then the third one is combinatorial delivery. So based on different types of pathways, you select the drugs and then put it together as a nanoparticle. So antibody functionalization and visualization, I said that antibodies can be used and they can take the medicine directly to the tumor, and that can help you in, it is also useful for diagnostics. Early diagnosis of the cancer can be done there. So the nanomedicine or nano trends are diagnosis. There are a lot of new diagnostic things are available, diagnostic nanomedicine. Then there is imaging. This is where you can use nanoparticles there, early detection of cancer, and finally drug delivery. So these are the 4 areas which are significantly impacted by the nanomedicine. You can stop me anytime. You are free to do that. So that is where the things come. So interesting approaches based on system. So please read this at home, because I have very less time remaining. But there are many different combinations are being used. So EFGR, HER2 combinations are inhibitions are there. They are trying to find out many different types of pathways and combining together using nanomedicine, using antibodies, they can resolve the cancer challenges. And this is where these approaches are going on in system biology. And you will see here now, this is your T cell which is protecting your body, and you can attach these T cells or get all these antibody, different types of things on the system and then deliver it. Now, if it can be targeted delivery, then you have less side effects. You will not see all the bad side effects which you normally see. And the last interesting thing which I am going to talk about is integrative medicine using herbal antioxidants and other natural products in combination with anti-cancer drugs is becoming a key. Now everywhere, if you come to Tampa, we have Moffitt Cancer Research Center, which is the third world's best cancer research center, and hundreds of clinical trials are going on there where they are putting herbal drug together with the anti-cancer drug, and I am going to show you some of those approaches today in the. So personal cancer therapy, you know, you can see the regorafenib is the new drug. Now it can work with many different pathways. You can have combination of bevacizumab, ZIP, antifabricate, sintuximab. Right? Now these are the combination of drug you are putting it, and angiogenesis and tumor microenvironment. So this is a very interesting area. It is really very fascinating to see that. And if I am lucky, in another 1 year, I am editing a book on tumor microenvironment and how to attack that so that rest of the body is protected from the drug. And we are working on that book proposal, and it will be in the print very soon. So there are different types of combination and combined therapies people are using. So you will find that you have different types of people, or sometimes people have good number of cancers. You know, I know a person identified as kidney cancer, has a gallbladder cancer, uterine cancer, and a lesion on pancreas. One shot in 1 year, all these things have happened. Now these are the things which can be challenging, and that's where the combination therapy is going to be very useful, and people are using that. Now precision medicine for cancer treatment, another area which is developing. There are a lot of researches which are going on, and you use different types of gene therapies. You know, particular cancer may be caused because of the one portion of the gene, so you can clip it, or you can neutralize it, or do something to that DNA so that your cancer treatment can be changed. So depends on where this part is. Now with the new technology and diagnostics, you can identify what gene is involved and which portion of the gene is involved for the cancer. And this is where the new technologies are coming into the picture. So now my last part, I have 5 more minutes, 10 more minutes. Oh, you are so kind. I'll finish before that. So now we'll try to find out how the herbal and nanomedicines are going to help. And that is where I would like to concentrate. So I don't want to repeat this. You know, World Health Organization has a herbal drug medicine. There are a lot of herbal drugs that are available. Indonesia has its own jamu here, very useful. There are indigenous herbal medicines, modified herbal medicines, and imported products on herbal medicine base. So many different things are there. There are supercritical extracts which are now readily available. You know Dexa Pharma, correct? Dexa Pharma. Have you heard this name? It's from America, correct? Dexa Pharma is from America. It's in Jakarta. Tangerang. I went to them. Yeah, they have developed many different herbal products. Very good product they have. I met their, you know, one of his vice president is a great guy. Raymond, you might have heard about him. So nanotechnology, I don't want to go into detail, but nano is too big. Lot of application all around the world. 10 raised to minus 9. And then term nanotechnology was used in 1974 by Norio Taniguchi. First time, this is the picture from my book, which tells you comparative study of the nanoparticles. So this is your hair follicle, RBCs, cells, and then this is a gene here. So nanofabrication, you all know it, top down or bottom up. So I am not going to repeat that. You must have heard about it. Nanotechnology was existing in the world for millions of years. But as we developed new techniques and technologies, we understood. So some of these techniques, transmission electron microscopy, dynamic light scattering, flow cytometry, ELISA, fluorometry, all these techniques are helpful in learning more and more about nanotechnology. Otherwise, we were eating grapefruit, or oranges, they were all nanoparticle. We drink milk, it is nanoparticle. So the challenges with the herbal drugs are high dose levels, consistency, reproducibility, clinical efficiency, manufacturing on large scale as a source of medicine for large number of people, disagreement between the practitioners and patient compliance. These are some of the challenges of herbal drugs, and nanomedicine can definitely help to resolve those. So we can reduce the dose levels for the herbal medicine, provide better stability to the product, ease in formulation, better patient acceptability, and it will help reproducibility. And then you can get the patents, intellectual property out of using nanotech. That's why I was saying that all these posters, if you convert it into nanoparticles and use the herbal natural Indonesian drug, you will get a patent. You will be able to patent that, and that is where you should start working, thinking about it, and creating your own companies also for that. So this is some of the very commonly used St. John wort, ginkgo, ginger, ginseng, curcumin, echinacea, chamomile. All these are in some or other ways recommended for different treatment, and a lot of clinical trials are going on, and people are they are part of the herbal drugs and nutraceuticals, or they are also called as botanical drug products. The US FDA is still not ready to accept that these products have therapeutic activity, so you cannot sell them as drugs. You have to sell them as nutraceuticals or herbal drugs. But these clinical studies are helping to understand how they can help you. Like valerian is useful in coronary heart disease. Like that, there are several things are there. Now, for herbal drug delivery, people are using liposome, polymeric missile, dendrimers, polymeric nanoparticle, solid lipid nanoparticle, and these are very easy to manufacture. These are very easy to prepare in your lab. You can do it in any lab. If any of you are interested, kindly communicate with me. In my PowerPoint presentation, I put my WhatsApp number as well as email address. Do communicate with me, and I will be able to help in how it can be done very easily. These are some of the polymers which have been widely used for creating such nanoparticles. So you have different types of synthetic, metallic, ceramic polymers which are used for nanomedicines, commonly used materials. And then you will find that there are several methods to prepare the things. So if one doesn't work, you can use the other one, but it is very easy. It is not very expensive to prepare nanoparticles in the lab. You just have to have passion and patience, both. Patience is necessary because first time it doesn't happen. It takes some time. For me, I work in the ophthalmic drug delivery, putting the nanoparticles in the eye, injection in the posterior eye. It took us 1 year to get a animal model to develop for macular degeneration. It took 1 year. It really takes a lot of patience, but you can't get anything without the patience. So these are some of the commonly used anti-cancer drug now. They are trying to find out how this can be utilized. So curcumin, there are a lot of clinical trials going on. Curcumin, lycopene, which is in tomato, tetradryan, transianon, tilbilene. So some of these things are now, people are seriously thinking about it. And there are many other herbal drugs which have been used for combining it with nanoparticles and putting them with anti-cancer drugs. And I will show you some of the patents which have been issued. So now there are new treatments developing. So they are having pulse electromagnetic fields for cancer treatment. Some alternative opportunities are happening. Oxygen, ozone therapy, ultraviolet blood irradiation, oxygen therapy, cold laser therapy, beamer therapy, hypothermia, ARP wave therapy, halo biophotonic therapy, liver flush, low dose chemotherapy. So these are some of the new techniques which people are using, even for people who are having tumors in the body, and they are finding some good applications of these things. These are not troublesome. These are much easier than the radiation as well as chemotherapy, and people working on it. So here there is the lymphatic massage to reduce the breast edema and swelling. This is one of the practice which is done quite widely in wellness. They call it wellness treatment systems, and wellness centers are there. Even Harvard Medical College, Stanford Medical College, all the top medical colleges, cancer centers nowadays have wellness center. Kindly go on, Google it, and you will find that all different types of technologies they are adopting for treatment of cancer. So Hercules multi-laser therapy, curcumin is widely used, IPT, mind, body, spirit meditation. This is a another very important part of wellness, because if your spirit is strong, if your mind and body is strong, you can fight cancer. If you say I am dying tomorrow, you die today. And that is where the mind body meditation is going to help, and this is very important thing. Then poly MBA, Healy, 25 plus IV therapy, frequency therapy, prescription, supplemental therapy, lymphatic therapy. These are some of the interesting alternative things which are going on. Ultrasound screening, acupuncture, massage therapy, cupping, nebulizer, curcumin, prolo therapy, laser therapy. These are some of the things which they are, they are now specializing in oncology massage. Can you believe that? But it is helping people. People are getting help from oncology massage. Why not? If that reduces your side effects, it is very useful, and that is where the things are coming. So nano drug delivery system is used for entrapping natural bioactive compounds for cancer and recent progresses. And so there are many different types of combinations of anti-cancer drug like cisplatin and curcumin. Now they have patent issued. Cisplatin and lipoic acid, cisplatin and glutathione. All these are natural antioxidants, because whenever you give anti-cancer drug, it creates a lot of inflammation in the body, like COVID. Why COVID people suffered a lot? Because it created a lot of inflammation in the body. And at that time, in many countries, they used herbal drugs, and COVID did not affect them much. And that is where the things were coming into the picture. So Ayurveda in India also is having some interesting cancer management system there. And one of the important thing is Sattva Vijaya counseling, then diet and lifestyle management. This helps a lot for the cancer patient. If you adjust your lifestyle, accept that you are a cancer patient and adjust your lifestyle, you may be able to live longer. And that is where, but you need the training for lifestyle management. And then there are nano drug delivery platforms are there. They are all different types of things people are using for delivering the drug for anti-cancer. So I have some of these US patents which have been issued where they have used the combination of astragalus, cisplatin and vinorelbine, cinnamonium, hilkinium and something. Then quercetin is also combined with the for human breast cancer. Bacillia oleracea L is combined with another cytotoxic drug and used in mammary carcinoma, cervical cancer. So now they are using antioxidant natural herbs in collaboration with but the cancer drug dose is reduced, and that is where you can find a lot of US patents. These are another US patents which are used. Curcumin is used for glioblastoma, the brain cancer. Curcumin and its analogs are used for pancreatic cancer. And all these clinical studies are going on. Probably in another 3 to 4 years, you will find these combination products available for cancer treatment with herbal thing. And yes, I am just completing. So please look at all this data which I have collected with you. And nano-based care solutions for oncology. So in the beginning, we started with the prices are going up. Now with the alternative therapies and herbal drug, the prices will go down. And that is where value-based oncology care is happening. You want to reduce the cost of expenses for cancer treatment and allow the person to live a little longer than in the normal case. And that's where phytochemicals at lower dose initiate adaptive response, but as one increases the dose, it produces acute autophagic response and apoptosis. So you can adjust the responses to the phytochemicals also. And I have seen some of the nice phytochemical studies in the posters. So a nano-based care solution for oncology is use nanomedicines, use herbal drugs, and have anti-cancer. So combination of these is new thing, but adopt all the alternative possibilities, acupuncture, massage therapy, oncology, massage, whatever you want to call it. But all these things are going to help to reduce the pain to the patient and reduce the side effects of the patient. So a lot to learn. I'm finishing. A lot to learn to be done. And I don't want to say this, but you can look at it, my slides. And my last slide is, my mentor, my boss, one of my good mentor told me that whenever you have more than 4 people in front of them, brag about yourself. And these are some of my books which I have published. So I go from nanoparticulate drug delivery system to nanotechnology, biointeraction, pharmacokinetics, and pharmacodynamics of these things. So there are many other books are there. I work in the nutraceutical area. Terima kasih.
Thank you so much for the amazing presentation. You can have a
Was
seat.
it bullet train or it was okay?
Yeah, you can have a seat, please. Okay. All right. We have come to the question and answer session, and maybe while waiting for the participants' question, maybe I will ask you some question. The first is, maybe can you tell us about the growth in nutraceutical use in America? Like here, we use the cancer therapy, and the nutraceutical is separated. And maybe before you talk about the combination of cisplatin and with the herbal, and here we have them separated, like cancer therapy and then the complementary from herbal. And maybe you can tell us about how is it going in USA about the nutraceutical usage.
Indonesia, doctors, they go in conferences, they talk that jamu is very bad. In the morning, they tell their wife, bring my jamu, I want to drink it. Am I right? Does it happen? Most of the people criticize that jamu, this, that, and all those things, but in the public forum, we need to change that. Jamu is very useful. Jamu has kept Indonesian people for thousands of years healthy. So, as a pharmacist, we should start standardizing the jamu so that there will be a reliability, reproducibility, and trust to the manufacturing. That is what we have to do. In America, as she has asked me the question, the anti-cancer, when it comes to the cancer, the people, there are a lot of experiments they do because people volunteer. They say that anyway, I am going to die in 3 months. Why not use my cancer as the experimental system? And that's why there are a lot of clinical studies are allowed by the FDA. So FDA, what it does is their style of working is that they call it investigative drug application, IND, investigative new drug application. So if I say that I want to use cisplatin and curcumin together, I will get the permission from FDA to use it. That is called investigative drug application, IND. And then the nutraceutical market is growing like anything, because as people are aging, people are finding difficulty, because if you go with modern medicine or so-called Western medicine or whatever you want to call it, it happens that you take a tablet and you get one disease relieved and create another disease in the body. You take something for diarrhea and then you have problems with your stomach, and more problems come. And that is why the elderly patients are saying that let me try the nutraceuticals or herbal drug. So the market, if my knowledge goes correct, the herbal and nutraceutical market in America was more than $186 billion. And you will see some of our say loudly. Yeah, so there are very good drugs here which are used in America quite widely, and these are jamu, they are jamu both, but they are excellent. So the American doctors are now changing their mindset. They are saying that if my patient is relieved with any of these treatment, I am ready to experiment. And that is where the mindset is changing. In Indonesia, it will change in another 5 years because we take a little bit more time. Once America does it, then rest of the world does it. So, but it is happening, and in Moffitt Cancer Research Center, hundreds of clinical trials are going on, combining antioxidants, anti-inflammatory to prevent the side effects of the drugs. So I think there is a good chance that a day will come, people will in around the world will talk about jamu. And I am one of them to talk about jamu. Everywhere I talk.
All right, thank you so much. You make me feel confidence to teach more about nutraceuticals because it keeps growing in USA. All right, maybe is there any question from the participants? Please raise your hand. You can ask both in Indonesia or in English. Okay.
You ask me in Bahasa and she will answer it in English. English.
That's Bismillahirrahmanirrahim. Good afternoon, Prof. Yaswan. I just make sure I'm not sleepy and I didn't sleeping during your presentation. So I asking some question for you.
Thank you. Terima
Okay,
kasih.
okay.
You have to say
It's
sama-sama.
my
I
name
say
Vidya.
terima kasih. You have to say sama-sama.
Oh iya, sama-sama. It's a good information about the nanoparticle topic. As we know, is the nanoparticle formulation have been used at the hospital in Florida. Maybe you can explain the clinical effectivity for the nanomedicine or nanotechnology because hospital at Indonesia haven't used this product yet. So how many percent the effectivity between nanomedicine or nanotechnology or conventional cytotoxic drug? That's it. Thank you.
You know, let me tell you now, as on today, if you go to American market, there are 45 plus products. They are nano products. Out of those 45 products, at least 25 to 30 are liposomes. Liposomal, correct. Liposome, liposome. Now, what is happening is these are specialized products. So they are doing the clinical studies and post-clinical study. Means they were applied as investigative drug. Now they submitted NDA, new drug application. They got FDA approved. But FDA has put a condition that these drugs and new nanomedicine drugs should be only used in cancer because they are specifically meant for cancer. And there has to be an oncologist who has to sign. So it is still a controlled market. It will take a lot of time because nanotechnology, I teach a course called risk management and nanotechnology. So there are a lot of risks involved in using nanoparticles. So the FDA is saying that we don't want to put nanomedicine in chemia pharma because the pharmacist who works in chemia pharma will not understand the importance of that medicine and usefulness of that medicine. So, and people are learning about it. So, the learning process is going on. People are using it. Only thing is all these 45 products are allowed only in cancer hospitals. There are some products that are used for other diseases, but again they are trying to restrict the use of it. Like cosmetics, you have cosmetics, correct? There are a lot of liposome cosmetics. If you go to France, almost a lot of nanoparticles are used in liposome. Now, if you know the skin protectors, you know, screen protective products, especially in sun, that sun protection products, people use nanoparticles. Now they found out that those nanoparticles, when they go into the sea, when you apply it on your body and go into the sea, that sea had corals, the corals were killed by these nanoparticles. Sea had certain fishes or certain animals which were killed by these nanoparticles. So in many, like in Hawaii, they have banned all the nanoparticulate skin protecting creams because they found out that it is an environmental challenge. So nanoparticle use is cautious. You have to be cautious to use it You have to find out, so it will take some more years to understand the side effect, understand the impact of environment, and whether it will be really good or not. Like fertilizers and insecticides, people use fertilizers and insecticides a lot. Now they are facing large number of people suffering from cancer. So people are now going towards organic farming. Because we did experiment, we failed, so we go back to natural things. So this is with nanomedicines will also happen. Am I answering your question? Did I answer your question or no? Terima kasih.
All right, sama-sama. Is there any more question? Okay.
Okay. Thank you, Mrs. Sabrina. Hello, Mr. Yasman Patak. My name is...
Professor.
Okay, just Patak only.
I work very hard to get being professor. I'm just joking. Go ahead.
Okay. Okay, my name is Malinda Prihandini. As you mentioned, Dexa Pharma. Actually, I used to work there before I pursue my master degree. Yeah. So my question now is about the herbal in the nanoparticle. Is it should be the single compound, or we can just put the whole extract, or maybe the fraction to the nanoparticle, and which one is more effective? Yeah, that's it. Thank you.
You know, if you want to create a product and sell in the West, good to have single particle because they don't understand. If you have to sell it in India, Indian people know it because Ayurvedic drugs contain 100 different herbal drugs. If you go to China, they understand that 100 different Chinese traditional medicine make it. So it depends on where you want to sell it. But normally what is happening is people tried. You know, I always give an example. When I did my PhD some 100, 200 years back, I'm joking. So at that time I used colorimeter. In those days colorimeter was the big thing. There was no spectrophotometer when I was student. So today if I submit my thesis for PhD, they will throw it in garbage. And I agree with them. I will also throw my thesis. But in 10 years back, it was very difficult to analyze certain drugs also. But then HPLC came. Now you can go up to nanometer analysis, correct? And now next stage is coming where people are analyzing picometer. So it is a matter of time that you will have analytical techniques where you can simultaneously analyze 100 ingredients of the herb. You know, curcuminides are 36 curcuminides. There are, if you take the analysis of curcumin, there are some 36 different ingredients are there. Yeah, all different types. So the new technology doesn't analyze it, doesn't mean that there will be no new technology. It is matter of time that in another 10 years, there will be new technologies to analyze, and then people will accept it that this is the way. But second way is that if we firmly say with some quality and all those things, then automatically, and another thing I want, shall I talk 1 minute? When is okay? Okay. You have to understand one thing. You play football, correct? So in football game, there are two teams, so one parameter. There is a ball, another parameter. There are ground, third parameter. There are two goals, fourth parameter. Like that you can have 10 parameters for football. Now in America, they play American football. Same parameters are there. But you cannot use the laws of American football to the regular soccer game. Correct? Because the games are different. So the herbal drugs, botanical drugs, jamu, Ayurvedic drugs, traditional Chinese medicines, the game is different. Western medicine, the game is different. You don't want to apply the rules of Western medicine to this traditional medicine because we will fail. But we will create our own game rules and say that, okay, this is my jamu, this is the surface tension, this is the viscosity, this is the drug content, and jamu is standardized. Now you are talking their language, standardization and creating appropriate reproducibility parameters. Once that is done, then people will accept it. I think we have good days. Don't worry. You will see those good days. I may not.
So it depends on where the place we gonna sell it, right?
Yeah.
All right. Okay. I think we have come to an end of this session, and ladies and gentlemen, I would like to take the conclusion from Professor Patek has presented today. So, nanotechnology plays an important role on cancer therapy, especially the targeted one that work specific on the cancer cell. And last, I'd like to thank Professor Patak for the amazing and informative presentation. And finally, give applause to Professor Patak for the presentation.
Terima kasih. Thank you very much.
So we have to say sama-sama. All right. Okay. All right. Thank you, everyone. So I will close this session. Wassalamualaikum warahmatullahi wabarakatuh.
Or not, for these 45 minutes, I traveled 72 hours. Thank you.
Disampaikan terima kasih kepada Ibu Apoteker Ayu Sabrina, Magister Farmasi.
Deliver thank you to Miss Apoteker Ayu Sabrina, Magister Farmasi.
Penyerahan sertifikat dan plakat kepada Profesor Yaswan Patak, Doktor Filosofi oleh Wakil Dekan 1, Ibu Doktor Apoteker Yulias Ninik Windrianti, Master of Science. Kepadanya disilakan.
Awarding of certificate and placard to Professor Yaswan Patak, Doctor of Philosophy by the first Vice Dean, Mrs. Dr. Apoteker Julius Ninik Windriati, Master of Science. Time is yours.
Partisipan yang berbahagia melanjutkan acara hari ini. Berikutnya adalah pemaparan materi dengan judul Nutritional Status of Croatian Student. Can we be satisfied? Oleh Zirinka Puharik, Medical Doctor, Professor Assistant. Pemaparan materi ini akan dimoderatori oleh Ibu Apoteker Junvidya Heroweti, Master of Public Health. Kepada Ibu Apoteker Junvidya Heroweti, Master of Public Health, disilakan.
I would not like to welcome our next invited speaker. We are extremely honored to welcome Assistant Professor Rinka Puharic, Medical Doctor Professor Assistant. This panel session will be moderated by Mr. Apoteker Junvidya Heroweti, Master of Public Health. The floor is yours.
Hi everyone. Can you hear me and see my presentation?
Yes.
Yes, okay. Well, I am very happy to see you again
Can you hear
and
me?
thank you. Yes. Can you hear me and see me? Is everything okay?
It's okay. It's so clear.
So, may I start?
Okay, let's start.
Okay. Dear Risha, Chairman of Organization Committee, thank you so much for invitation. I'm honored and pleased to be here today with you.
Okay.
My name is Rinka Puharic. I'm medical doctor, professor assistant and dean in University of Applied Sciences Bjelovar in Croatia. And my special fields are public health,
I'm sorry,
epidemiology, and
Zrinka.
dietetics. Sorry?
I opened the conference first and then you join. It's okay?
Okay, can you hear me and my presentation too?
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Honorable all the participants and of course all invited guests. Good afternoon all. My name is Junvidya and now I am your moderator at the international conference. It's so awesome and precious chance for me. And I hope you still have enthusiasm to listen the conference because I know this is the critical hour. You're not sleepy? I'm sorry, mister. This is the critical hour, but trust me, this material or this topic is so awesome, or so maybe you can join this topic. And for the conference, we have a speaker who already with us, Zrinka Puharic, medical doctor, professor, assistant. And I like to greet Zrinka. Good morning, Zrinka.
Good morning.
Yeah, as we know, Croatia is 5 hours faster than Indonesia, so they're at 9 a.m., isn't right?
9 a.m.
Yeah, in here still 3 p.m., Zrinka. How are you, Zrinka?
Very well, thank you. How are you?
I'm good. Okay, our presentation around 20 minutes, and then we'll have a discussion for about 40 minutes. And for your information, Zrinka, our audience are faculties, researcher, practitioner, and student also. So quite various background. And all the audience, maybe you can ask a question during the discussion session by raising your hand. Is it right? Okay. And now I'm going to invite Zrinka. I'll read your profile. Zrinka Puharic, medical doctor, professor, assistant. She is Dean of the University of Applied Science Bjelovar and teaching public health, hygiene, and epidemiology dietetics. And on May 28, 2018, she was elected to the scientific teaching title of assistant professor in the scientific field of biomedicine and health in the field of public health and healthcare. And I have noticed a lot of paper for you, Zrinka. All right, today Prof. Zrinka will present about the dietary habit for cancer patient. As we know that cancer patient had a decrease in appetite, which leads to weight loss and nutritional status due to the cancer itself or the effect of the therapy. So maybe Zrinka, you have 20 minutes to present your presentation. So please.
Thank you so much for your introduction, for your wonderful words. Thank you so much, Risha and other friends, to invite me. I'm honored and pleased to be here today. It's 9 a.m. in the morning in Croatia, so I'm very fresh to talk about the nutritional status of Croatian students. Can we be satisfied? Well, on this slide, you can see that European Union has huge problem with overweight and obesity population. BMI is calculated as person's weight in kilograms divided by the square of height, and average for European Union is 53%, which means that every second person in European Union is overweight or obese. Croatia is also above this average with 65% of overweight and obese person. If you're looking by age, we can see that highest years means more percent of obesity till the 75 years and over, which is expected fact because of the body changes and less work activity. Maybe we must be worried about this slide, which shows overweight and obesity of children. We could see red zones, a Mediterranean country plus United Kingdom, Croatia also. We all know that obesity in childhood can lead into obesity for whole life, so it's extremely important to keep children normal weight. Nearly 1 from 6 European adults is considered obese, average is 15.9%, and the most obese country in European Union are Malta, Latvia, Hungary, Estonia, United Kingdom, and Czech Republic. The state which the least obese person is Romania, Italy, Netherlands, Belgium, Sweden, Cyprus, and Austria. You can see all European states on this slide. According to the boys and girls, you can also see European Union countries with the most obese children. It's very surprising that it's a Mediterranean country because we expected that they are eating more fish, more olive, and so on, more fruits and vegetables. But the most obese boys in European Union are in Cyprus, Italy, Greece, Montenegro, San Marino, and also the girls is from Cyprus, Spain, Malta, Greece, Italy, and from Macedonia. What about creation data? I think maybe I skip some. No. Creation data says that increased body weight was registered in 12% of primary school students. It means the children between 6 and 14 years old. In 14% of secondary school students, it means 14 to 19, and in 16% of university students. Also, obesity was found in 17% of primary school students, 12% of secondary school students, and 5% of university students. What is cause of obesity? Of course, too much intake calories, and we are not moving so much as the scientific recommendations are. So in European Union, we eat too many cured meats, and we all know that cured meat is full of addictive substances which our human body can't digest. We also here in Europe drink too many sweetened juices and alcohol full of hidden calories, which is not good for our body and sugar level. Of course, we are too much in front of the TV. We are sitting and we are spending more than 7 hours per day watching in our glow screens. World Health Organization says that we can walk at least 30 minutes or 10,000 feet per day, but let's be honest, most of us do not do that every day. Let's talk about my little research. Aim was to investigate dietary habits of university students. We are a small private university. We have 3 departments: mechatronic, nursing, and computer science, all from STEM fields. And we have about 800 students. It means that almost 65% of all participants wanted to volunteer in this research. The study involved a total of 500 respondents. The most was female students, 361, and 139 was male. Average age was 25 years, 23 was from the city, and 77 was from surroundings area. Of course, we have a proof of ethical committee, and we used anonymous questionnaire. And for the fat and muscle tissue, we use the SECA scale. You can see it on the screen. We measured height and weight and body mass index for our students in research, and we discovered that we have 7% of underweight students. We have 57% of normal weight students, 26% of overweight, and 10% of obese students in our university. When we split our participants into male and female, we don't find statistical difference between male and female. in this 4 category: underweight, normal weight, overweight, and obese. And also when we split them into village or surrounding area and the city area, there is no statistical significance between our participants. When we measure the fat tissue, we discovered that half of our students have high and over % of fat according to the recommendations. Also, when we measure the muscle tissue with our students, we found that 11% have less level of muscle tissue, 58% normal, 27% overweight, and 3% is very, very high % of muscle tissue in the body. Of course, we use anonymous questionnaire to examine what our students eat and what kind of food may affect on fat and muscular tissue for our students. We ask a lot of questions about dietary habits. Here are some important facts. In Croatia, the most used fat is margarine and sunflower oil. Only 10% eat vegetable every day, and only 12% eat fruits every day. We all know that the World Health Organization recommends 5 meals per day. Also, 62% of participants does not drink milk at all, and 55% of participants eat bakery products every day. It's something like Croatian standards. We have bakery in every corner, so especially near primary and secondary schools, so our children eat bread and bakery products so much in Croatia. This is the slide when we try to find what kind of food may affect on fat and muscular tissue. And we agreed that consumption of cereals has no effect on the proportion of adipose tissue. Consumption of potatoes and sweets significantly affects the percentage of adipose tissue of male students. Also, fruit consumption has a positive correlation on the body proportion of adipose tissue in both groups of respondents. Increased intake of milk and dairy products increase the proportion of adipose tissue in female students. Alcohol intake increased the proportion of adipose tissue in male students. That we can conclude with statistical significance. Of course, we consumed a lot of meat in Croatia, fish and eggs, which increases the share of muscle tissue in both groups of students. And consumption of milk and dairy products increases the share of muscle tissue in male students. Conclusion: can we be satisfied? After this research, we can say that Croatian students are like any other European Union students, according to the percentage of obesity and overweight and dietary habits. What about sugar and refined carbs? Processed foods that are high in sugar and low in fiber and nutrition have been linked to a higher cancer risk. In particular, researchers have found that a diet that causes blood glucose levels to spike is associated with an increased risk of several cancers, including stomach, breast, and colorectal cancers. One study in over 100,000 adults found that those who consumed a diet high in refined carbs were almost twice as likely to die from colon cancer than those who ate a diet low in refined carbs. It's thought that higher levels of blood glucose and insulin are cancer risk factors. Insulin has been shown to stimulate cell division, supporting the growth and spread of cancer cells and making them more difficult to eliminate. In addition, higher levels of insulin and blood glucose can contribute to inflammation in your body. In the long term, this can lead to the growth of abnormal cells and possibility contribute to cancer. This may be why people with diabetes have an increased risk of certain types of cancer. risk of colorectal cancer is 22% higher if you have diabetes. Of course, to protect against cancer, limited or avoid foods that boost insulin levels, such as food high in sugar and refined carbs. What about processed meat? And we'll eat a lot of processed meat in Croatia. The International Agency for Research on Cancer seems that processed meat is cancerogen, substance that cause cancer, because processed meat refers to meat that has been treated to preserve flavor by undergoing salting, curing, or smoking. It includes hot dogs, ham, bacon, salami, and some deli meats. Observational studies have found association between consuming processed meat and increased cancer risk, particularly colorectal cancer. A large review of study found that people who ate large amounts of processed meat have a 20 to 50% increased risk of colorectal cancer compared to those who ate very little or none of this type of food. Some observational studies have also linked red meat consumption to an increased cancer risk. What about overcooked food? When you cook in certain foods at high temperatures, such as grilling, frying, broiling, and barbecuing, we can produce harmful compounds like heterocyclic amines and advanced products. Excess buildup of these harmful compounds can contribute to inflammation and may play a role in the development of cancer and other disease. Certain foods, such as animal food, high in fat and protein, as well as highly processed food, are most likely to produce these harmful compounds when subjected to high temperatures. These include meat, cheese, fried eggs, butter. I mentioned margarine in Croatia, cream cheese, mayonnaise, oils, and nuts. To minimize cancer risk, avoid burning food and choose gentler cooking methods, especially when cooking meat, such as steaming, stewing, or boiling. And what about the four food products, dairy products? Some observational studies have indicated that high dairy consumptions may increase the risk of prostate cancer. One study which followed almost 4,000 men with prostate cancer, and the result shows that high intakes of whole milk increases the risk of disease progression and death. Theories suggest that these findings are due to an increased intake of calcium, insulin-like growth factor, or estrogen hormones from pregnant cows, all of which have been weakly linked to prostate cancer. Also, when you overweight or obese is linked to increased cancer risk. We now know the 13 different types of cancer, including esophagus, colon, pancreas, and kidney, also the breast cancer after menopause. Obesity can increase cancer risks in 3 different ways. Excess body fat can contribute to insulin resistance. Obese people tend to have higher level of inflammatory cytokines in their blood, which causes chronic inflammation and encourages cells to divide. And fat cells contribute to increased estrogen level, which increases the risk of breast and ovarian cancer in postmenopausal women. So what we can do as public health workers? In that student age, which is average 24, actually not so much. Preventive programs and workshops can be prepared for kindergarten children and their parents. It's time for forming knowledge and attitudes. So we must act from the childhood by promoting consumer awareness of healthy diet, developing school policies and programs that encouraging children to adopt and maintain a healthy diet, educating children, adolescents, and adults about nutrition and healthy dietary practices, encouraging culinary skills, including in children taught schools, and providing nutrition and dietary counseling at primary health care facilities. Thank you so much for your patience.
So is there any question? Maybe we discuss about the obesity is also a cause of case of disease such diabetes or a cancer. Is there any question, maybe? Okay.
Okay, hello Zrinka. For me, my name is Malinda. Okay.
Hello.
Hello, how are you?
Very well, thank you. You?
Okay. Okay, Zrinka. And that as is dietary maintenance working to support the cancer therapy in Croatia? Is there any specific dietary for the patient for of the or because in Indonesia maybe it's just yeah, I think it's not been conducted yet. Thank you.
Thank you for your question. Thank you for
the
your
point,
question.
Zrinka?
Do you get the point?
Yes, I get the point. Unfortunately, I'm not the oncological, but I know that it depends on the kind of cancer. Every clinical hospital in Croatia have a dietetic specialist, and they show the patient, they write to the patient the dietary. It depends on the kind of the cancer, if I understand correctly the question.
Okay, so maybe if the adequacy of food from outside is not qualified, maybe we can give a parenteral nutrition or how the protocol at a Croatia. Because in Indonesia, the nutrition or parenteral nutrition not conducted so well.
We have some recommendations that the clinical doctors can write, and they are writing for some cancers, and you can follow these nutritional procedures. And also we know that you are what you are eating for, so it's very helpful to treat the cancer.
Okay, thank you. Is so clear enough, Mrs. Malinda? Okay, thank you, Zrinka. Is there any question? Okay.
I hope you can hear me. I have a question. Did they have Croatian traditional diet systems which have been altered leading to the obesity increase, or they were practicing the same traditional diet what they are doing now?
Can you get the point?
Can I repeat again?
Please.
Repeat it.
Yeah. Was there any traditional diet system in Croatia which deviated leading to the obesity now?
Well, in my country, when your body mass index is over 30, which means that you pass the border to the obesity, you can go to the clinical hospital, and for free, the clinical doctors will resolve your problem. It includes medicine, the dietary advisor, maybe some clinical points, and they find to try the reason. Is it your dietary habits? Is it your hormones or something else? And they try to solve your problem in the clinical hospital in the main town.
Okay, thank you, Zrinka. Is there any question? Okay, Zrinka, may I ask you, what is a good food composition for a cancer patient? Like how many percent the carbohydrate, the fats, or the protein, or maybe you have a good diet strategy to cure the cancer patient?
Well, unfortunately, we in Croatia don't have a very good strategy to cure the cancer, but we are trying to. As I said previously in my presentation, please avoid the meat with also lots of products in the meat. Avoid alcohol, hidden sugar, and of course carbohydrates. I think that in European Union we live very fast, so we can't cook healthy meat. So we eat in the bakery. We use a lot of fast food, which is not very good. We see on our slides the rate of cancer is higher and higher every year in European Union. It's a serious problem, and we must figure out how to resolve that.
Okay. So the healthy food to contribute to cure the cancer patient. Is it right?
Yes.
Okay. Is there any question? Is enough? Okay, it's enough, Srinka. We finally come to the end of the conference. So I would like to take the conclusion. Providing balanced nutrition is very much needed in cancer patients so that patient can get adequate nutrition. I'd like to thank you to the speakers, Rinka Puharik, for the informative and the interesting presentation, and for all your participants. Finally, give applause for the speaker and for you all. Thank you. Thank you, Rinka. Assalamualaikum warahmatullahi wabarakatuh.
Disampaikan terima kasih kepada Ibu Apoteker Junvidya Herwati, Master of Public Health.
Deliver thank you so much to Mrs. Apoteker Junvidya Herwati, Master of Public Health.
Berikutnya adalah pengumuman pemenang Best Oral Presentation dan poster terbaik. Best Oral Presentation dari Ruang 1 adalah Fahri Mubin. Kemudian ruang 2 adalah saudara Eva Monika. Namun karena saudara Eva Monika tidak ada di tempat, maka pemenangnya adalah Apoteker Yance Anas, Master of Science. Sedangkan pemenang presentasi oral secara online terbaik adalah saudara Denny Satria. Dan pemenang poster terbaik adalah Apoteker Devi Nisa Hidayati, Master of Science. Kepada Fahri Mubin, Apoteker Yance Anas, Master of Science, Apoteker Devi Nisa Hidayati, Master of Science, dipersilakan untuk dapat maju ke depan untuk menerima sertifikat. Kepada Wakil Dekan 1 Fakultas Farmasi Universitas Wahid Hasyim, Ibu Dr. Apoteker Yulias Ninik Windriati, Master of Science, mohon berkenan menyerahkan sertifikat kepada para pemenang.
Next is the announcement of the winner for the best oral presentation and the best poster. The best oral presentation from room 1, 2, and the best online oral presentation are Fahri Mubin, Apoteker Yance Anas, Master of Science, Denny Satria, respectively. And the winner of the best poster is Apoteker Devi Nisa Hidayati, Master of Science. To Fahri Mubin, Apoteker Yance Anas, Master of Science, Apoteker Devi Nisa Hidayati, Master of Science, are welcome to come forward to receive a certificate. To the first Vice Dean, Mrs. Dr. Apoteker Zulias Ninik Widriati, Master of Science, please give the certificate to the winner.
Kepada saudara Denny Satria sebagai peserta presentasi oral terbaik, sertifikat nantinya akan dikirimkan. Bapak Ibu peserta seminar, dengan berakhirnya sesi yang dibawakan Asisten Profesor Zrinka Puharik, spesialis, maka berakhir pula acara hari ini. Maka mari kita tutup acara ini dengan bacaan hamdalah bersama-sama. Alhamdulillahirabbil alamin. Terima kasih atas partisipasinya. Kami mohon maaf bila ada hal yang kurang berkenan di sepanjang jalannya acara hari ini. Sampai jumpa di kesempatan berikutnya.
Ladies and gentlemen of the seminar participant, with the end of the session, brought by the Assistant Professor Zrinka Poharic, Medical Doctor Professor Assistant. Then the event ends today. Thank you, your for participant. We apologize if there are things that are not pleasing along the course of today's event. See you on the next event.
Wassalamualaikum warahmatullahi wabarakatuh.
Oke, pengumuman kepada para peserta yang ada di dalam